Good afternoon, everyone. I'm Catherine Sewell, Deputy Director for Safety in the Division of Urology, Obstetrics, and Gynecology in the Center for Drug Evaluation and Research at the FDA. Welcome to our public meeting, Scientific and Ethical Considerations for the Inclusion of Pregnant Women in Clinical Trials, which is being convened through a cooperative agreement between the FDA and the Duke-Margolis Center for Health Policy. Throughout the day, we'll have a series of presentations, reactant panels, and moderated discussion sessions covering approaches to the promotion and conduct of clinical studies in pregnant people. We are joined by a group of subject matter experts listed with speaking roles on the agenda, and also several hundred stakeholders who've registered for this meeting. We're very glad that each of you is able to join us today. May I have slide 3, please? First, I'd like to go over a few reminders about our meeting format to help us organize discussion in the virtual setting. The meeting materials are available on the Duke-Margolis website. We also have Twitter information on the website, and participants can feel free to tweet about the meeting using the hashtag #ResearchInPregnancy. To our workshop speakers, Kaylee Probst will advance the slides on our end, so please give a verbal prompt that you'd like the next slide. Additionally, if you would take a moment now to update your participant chyron with your affiliation, that would be helpful. If you're unable to do so, Duke-Margolis can help with that. If you would like to speak during the open discussion portion of the meeting, either during your session or during other sessions, please switch your camera on and raise your virtual hand by clicking on Participants and then clicking on Raise Hand. Please keep yourself muted when you're not speaking. If you have a question or comment during a presentation or during the open discussion, feel free to type it into the Zoom chat box and address it to all panelists and attendees. To our registered stakeholders, if you would like to ask a question or make a comment during the open discussion portions of the meeting, please type it into the Zoom Q&A box. We have a packed agenda today, and to ensure that we wrap up on time, it would be great help if each of the presenters would keep their remarks to the allotted time limit, and if each of the panelists would keep their remarks to 5 minutes before each panel discussion. If you have technical issues, please send a direct Zoom message to Rashid Willis or email rwillis@newmediamill.com. There is live closed captioning available today. A recording of the meeting will be available on the Duke-Margolis event website and YouTube channel at the conclusion of the event. The Duke-Margolis Center is convening today's meeting in cooperation with the FDA. This is not a federal advisory committee meeting. We're keenly interested in your input on this topic, but we will not be following advisory meeting procedures or taking a vote at the end of the meeting. The workshop will be a success if there's a vigorous exchange of ideas and open discussion. One final housekeeping note, I would like to remind everyone that we aspire to use inclusive language during this meeting. I acknowledge that several US government documents, including the Federal Register Notice, guidances, and regulations, use the word women. FDA recognizes that sex and gender are distinct terms, with sex being defined as a biological construct and gender as a social construct. FDA understands that people self-identify by gender, and for some, gender and sex are concordant, while for others they may not be. Discussion of this topic falls outside of the scope of this meeting. However, our speakers and panelists do aim to be as inclusive as possible. I would also just like to take a moment to thank the staff at Duke-Margolis, in particular Sarah Sheehan, the Managing Associate for Biomedical Research and Clinical Trials, Research Assistants Mira Gill, Haley Sullivan, and Kaylee Probst, Morgan Ramin, Chief of Staff, Dr. Marta Woshinska, the Deputy Director for Policy, and the many others who are coordinating this virtual event today. I would also like to thank my colleagues at the FDA who've worked so hard to plan this event. Those colleagues from the divisions of Urology, Obstetrics, and Gynecology, Pediatrics and Maternal Health, Pharmacology, Toxicology, Anesthesiology, Addiction, and Pain Medicine, and those colleagues from the offices of Women's Health, Pediatric Therapeutics, Good Clinical Practice, Pharmacology, and Infectious Diseases, as well as our public meeting support and communication staff. Special thanks to those of us who played a central organizing role. You have worked tirelessly for almost 2 years to bring this workshop to fruition. So on to the focus of our workshop. Next slide, please. As everyone who's here today understands, pregnant people have historically been excluded from clinical trials for new and existing drugs, but many of them still use medically necessary drugs during the course of a pregnancy. Clearer scientific understanding of the risks and benefits of using medications for pregnant people and their developing fetuses is essential to the safe and effective management of pregnancy-related conditions, as well as chronic and acute medical conditions experienced during pregnancy. Today, we'll have 3 sessions. In session 1, we'll hear from presenters about the need for and existing guidance for the participation of pregnant people in clinical trials. We'll also hear from our NIH colleagues about the status of implementation of the PregLac Task Force recommendations, and we'll close out the session with a clinician's perspective on the evidence gap for therapeutics prescribed during pregnancy. In session 2, we will have participants address non-clinical safety assessments to support clinical trial enrollment of pregnant people, including characteristics of a standard non-clinical program to support the safety of a product in the antenatal period. And finally, in session 3, we will discuss scientific and ethical considerations when designing clinical trials that enroll pregnant people, including the therapeutic context and the timing of data collection. Tomorrow, we'll bring it home with case studies and a session on the challenges and actionable next steps. I'm confident that these will be engaging and exciting discussions, and I'm hopeful that our conversations can facilitate drug development to address unmet medical needs in the pregnant population. I would now like to turn it over to Dr. Kavita Vasish, Associate Commissioner for Women's Health and Director of the Office of Women's Health at the FDA, for her opening remarks. Kavita.
Thank you, Catherine. Can you go to my first slide, please? Next slide. And next slide. These are our standard. Okay, that's fine. Those were my disclaimers, and I have no conflicts of interest. I just want to thank Catherine. It's really a pleasure for me to be here today to talk to you on behalf of FDA's Office of Women's Health and to really briefly touch on FDA's work to support pregnant and lactating individuals. I thought I'd begin by first talking a little bit about our office. We were established over 25 years ago by congressional mandate in the Office of the FDA Commissioner. And our mission is to promote the inclusion of women in clinical trials and the implementation of guidelines concerning the representation of women in clinical trials and the completion of appropriate sex and gender analyses. We also identify cross-cutting and multidisciplinary women's health initiatives, including changing needs, areas that require study, and new challenges to the health of women as they relate to FDA's mission. And we serve as the principal advisor to the commissioner and other key agency officials on scientific, ethical, and policy issues relating to women's health. We do this work by working in 3 key areas: science, education, and engagement, which includes our outreach and communication efforts. Next slide, please. In alignment with our mission, our Office of Women's Health serves as the FDA Commissioner's representative on the task force on research specific to pregnant and lactating women, also known as PregLac. The 21st Century Cures Act established PregLac to advise on gaps in knowledge and research on safe and effective therapies for individuals who are pregnant and or lactating. Maternal health is very important. It's critically important focus area across FDA. Therefore, we created an internal FDA work group with representation from product centers as well as other offices to provide a multidisciplinary and comprehensive perspective from FDA to inform the work of the PregLac Task Force, including the initial PregLac report, which was published in 2018, and the follow-up implementation plan report, which was recently published. Next slide, please. In addition to our work on the task force, FDA has several activities and initiatives that aim to support our public health mission related to pregnancy and lactation. And this includes efforts to advance policy, research, outreach, and education. And I'll be touching upon these just very briefly in the next few slides. Next slide, please. So from a scientific and ethical standpoint, the complex interdependency of maternal and fetal well-being should be taken into consideration when assessing the risks and benefits of medication taken during pregnancy and lactation. In 2019, the FDA put out the first in a series of draft guidance documents, which discusses the agency's perspective and recommendations on those scientific and ethical issues that should be considered in clinical trials supporting drug development. This was followed by the draft guidance on post-approval pregnancy safety studies, which provides sponsors and investigators with recommendations on how to design investigations to assess the outcomes of pregnancies when people are exposed to drugs and biological products regulated by FDA during a pregnancy. And the agency also put forth guidance, which provides recommendations for how and when to conduct clinical lactation studies and how to assess the influence of biologics or drug products on lactation. Later this morning, you will hear from my colleague, Dr. Layla Shaheen from the Division of Pediatric Maternal Health, who will further expand on FDA policy, including some of our more recent guidance. Next slide, please. Since the Office of Women's Health's inception over 25 years ago, we've worked across FDA offices and centers to support research studies on a broad range of topics, including pregnancy and lactation. A few years ago, we published FDA's first Women's Health Research Roadmap, and this roadmap really incorporated cross-agency input to create strategic direction by outlining priority areas to really help us maximize the impact of our research initiatives and ultimately to promote optimal health for women. I've outlined on this slide some of our more research projects that we funded over the past few years. And many of these projects represent intramural research conducted by our own FDA staff. We also have funded extramural research through our Center for Excellence in Regulatory Science mechanism and our broad agency announcements. And I won't go into detail here, but I just wanted to highlight the diversity of the projects, ranging from looking at real-world use of pharmaceuticals among pregnant and lactating individuals to using in vitro and to in vivo extrapolation in human breast milk, looking at how the PLLR, the Pregnancy, Lactation, and Labeling Rule, and how health communications around that rule, and also supporting funding around an artificially intelligent virtual pregnant woman. And our research program is really a mechanism through which we aim to expand our collective knowledge and bridge important data gaps. Next slide, please. So FDA and NCATS have built a mobile application and website called Cure ID, and this is to capture experiences from clinicians around the globe when they find novel ways of using FDA-approved drugs to treat infectious diseases, emerging threats, and multi-drug resistant organisms. This past year, the Office of Women's Health recently funded expansion of this program to collect data on the use of drugs for infectious diseases and cancer during pregnancy. Next slide, please. So I've highlighted our research program. I also wanted to mention that we on our OWH, we call ourselves OWH for the Office of Women's Health, our website, we maintain a listing of pregnancy exposure registries. So a pregnancy exposure registry is an observational study. It gathers health information from individuals who take a medication or receive a vaccine during pregnancy. And these registries are one way for us to get information about maternal and fetal effects of medication exposure during pregnancy. We maintain this website as really a valuable resource for potential participants as well as healthcare providers. There are about 120 medical product registries on the website now, which link to drug information, as well as patient education resources. And I have the website here for you because I think it's important for people to know that this is available if we really want to encourage participation in clinical trials. Next slide, please. FDA also recognizes the need for coordinated research to address public health needs during the perinatal period. So this includes the health of the mother, premature infants, and newborns, as well as development throughout childhood. And so to that end, our National Center for Toxicological Research established FDA's virtual Perinatal Health Center of Excellence, which we call the PHCE. The PHCE was essentially created to focus on these populations and increase opportunities for collaborations, leveraging research opportunities, and to support research that applies to the understudied populations and within this period, perinatal period, which I just described a little bit about before. Next slide, please. So education is also a very important mechanism for us to get our word out and to also bring in people to educate us on really important critical topics. So we have developed a scientific speaker series, and this aims to bring in world-renowned experts to share the latest discoveries with our FDA staff and federal partners. These lectures really focus on emerging and critical topics, such as our seminar focused on COVID-19 in pregnancy, which was held in August. In addition to these quarterly seminars, we also host workshops and webinars. So in 2019, we hosted a meeting on the safety of asthma medications in pregnancy. And just this past November, we held a multidisciplinary scientific conference on CBD and other cannabinoids, sex and gender differences in use and responses. And this conference was really focused on highlighting the existing research as well as knowledge gaps. And part of this full day conference included one session, which was exclusively focused on discussing the use of CBD and other cannabinoids in pregnancy. So in the process of developing the meeting report, and that will be available on our Office of Women's Health website. Next slide, please. I also wanted to share that our office offers easy to read free publications on a variety of health topics. And this is really our FDA's way of engaging with our stakeholders and outreach. You can download and order these free copies. We have about 40 fact sheets and brochures in multiple languages. And as depicted on this slide, we offer many, many resources that pertain to pregnancy. So I encourage you to look at these resources as well. And next slide, please. Finally, I'd like to invite you to stay connected with us. You can follow us on Twitter, where we often tweet about our resources. We often use this as a way to encourage people to participate in clinical trials and to promote awareness around our pregnancy registry webpage. You can also sign up for our Women's Health Monthly newsletter, which really serves to disseminate very important information from the agency pertaining to women's health. Next slide, please. So thank you very much, and I'm really looking forward to the meeting today.
Thank you so much, Kavitha. This is Susan McCune. I'm just waiting to get my video up. I apologize. So I just got a chat that everyone can see me, which is great. So thank you very much. And thank you, Kavitha, for an excellent talk. My name is Susan McCune. I'm the director in the Office of Pediatric Therapeutics in the Office of Clinical Policy and Programs in the Office of the Commissioner at the FDA. I will ask everyone's indulgence and patience with me. I'm much better in the non-virtual space, although I have to say I'm glad that we are virtual today, given the fact that we had to cancel a pediatric advisory committee meeting because of snow in the past. So when I saw the snow, I was actually pleased that we were already all in the virtual space. So I'm going to be your moderator for the first session today, and we're going to run till about 1 o'clock, and then we'll turn it over to the second session. In the first session today, this is entitled Understanding the Need and Existing Guidance for the Participation of Pregnant People in Clinical Trials. Our panel of experts will cover 3 different areas. The first is to discuss regulatory, scientific, and ethical considerations for enrollment of pregnant people in clinical research as articulated in FDA guidance. Secondly, to discuss objectives of the PregLac, the accomplishments and future goals. And then thirdly, to discuss urgent and key areas of unmet needs for therapeutic development or clinical data in obstetrics. So we will have a series of presentations, first from Layla Shaheen, who is a senior medical officer in the Division of Pediatric and Maternal Health in CEDAR at FDA. And Layla will be followed by Erin Powlik, who is the Chief of the Obstetric and Pediatric Pharmacology and Therapeutics branch at the National Institutes of Health. And then Erin will be followed by Jean Sheffield, who is the Division Director of Maternal Fetal Medicine in the Department of Gynecology and Obstetrics at Johns Hopkins. So first up is Layla Shaheen. Layla.
Thanks, Susie. Good afternoon, everyone. Thank you for your interest and for participating in this workshop. Next slide, please. Next slide, please. So this presentation represents my personal views and not the official position of the FDA. Next slide, please. So the objectives of my talk are to discuss FDA efforts to advance clinical trials in pregnant people, review the regulatory framework for pregnant people in clinical trials, and discuss FDA efforts to collect data in pregnant and lactating people with COVID-19. Next slide, please. I think it's important to start off by stating that FDA is committed to advancing research in pregnant and lactating people. We need data to inform prescription drug labeling and to inform benefit risk considerations in prescribing for pregnant and lactating people who have long been underrepresented in clinical research compared to the general adult population and pediatrics. But there's growing understanding of the need for an earlier and more systematic approach to studying pregnant and lactating people. FDA has demonstrated its commitment to pregnant and lactating people by publishing several guidances for industry in the last few years, some of which are dedicated to pregnant and lactating people, and others that are related to the general population but highlight issues related to these populations. Regulatory advances have occurred. We have actually come a long way. Until recently, federal regulations used to lump pregnant people in with children, prisoners, and individuals with impaired decision-making capacity. Stakeholders felt that this was paternalistic and that pregnant people can certainly make informed consent decisions for themselves. So the common rule has removed reference to pregnant people as vulnerable, and FDA is working to harmonize its own regulations with the common rule. Additionally, FDA is a participant on the PregLac task force and is committed to working with NICHD and other stakeholders to address the data gaps in pregnant and lactating people. Next slide, please. The Pregnant Women in Clinical Trials guidance was published in 2018 and is important because it is an endorsement of the need to include pregnant people in clinical research, and it aligns with the PregLac recommendations. Next slide, please. The guidance discusses the ethical and scientific considerations for when it would be appropriate to include pregnant women in clinical trials, and follows the HHS framework of human subject protection regulations. The guidance discusses considerations for the post-market versus the pre-market setting and how to handle women who become pregnant during a trial. FDA is reviewing the public comments and will consider key messages and input received from this meeting. Next slide, please. Federal regulations 45 CFR Part 46, Subpart B, include additional protections for pregnant women, which includes 10 specific requirements. These regulations apply to research that's conducted or supported by HHS, however, are recommended by FDA and are generally followed by stakeholders. I'm going to go through all 10 requirements as they provide the basic framework for the conduct of clinical trials in pregnant people. The first requirement is that where scientifically appropriate, non-clinical studies, including studies in pregnant animals, and clinical studies, including studies in non-pregnant women, have been conducted and provide data for assessing potential risks to pregnant women and fetuses. Next slide, please. The second requirement is that there needs to be a prospect of direct benefit to either the woman or the fetus. If there's no prospect of direct benefit, the risk to the fetus must be minimal, and the purpose of the research is the development of important biomedical knowledge that cannot be obtained by any other means. Minimal risk is defined in the regulations as the following: the probability and magnitude of harm or discomfort anticipated in the research are not greater in and of themselves than those ordinarily encountered in daily life or during the performance of routine physical or psychological examinations or tests. This minimal risk requirement was identified as a barrier to research by the PregLac task force, which recommended that there be a consideration for a minor increase over minimal risk similar to pediatric regulations, and that experts and stakeholders should be convened to reach consensus prior to making any revisions to this requirement. Next slide, please. The third requirement is that the trial involve the least possible risk for achieving the objectives of the research. The fourth requirement is that informed consent is obtained as described in Subpart A of the regulation. The fifth requirement is as follows. If the prospect of direct benefit is solely for the fetus, then additional consent from the father is needed unless he's unavailable, incompetent, has temporary incapacity, or the pregnancy results from rape or incest. The PregLac task force found this requirement to be a regulatory barrier as well, as family structures have changed and this requirement seems paternalistic and outdated. Next slide, please. The next 5 requirements are pretty straightforward. Participants are fully informed of the reasonably foreseeable impact of the research on the fetus or the neonate. For children who are pregnant, assent and permission are obtained. There can be no inducements for pregnancy termination. Investigators cannot be involved in decision-making regarding pregnancy termination, and investigators cannot be involved in determining the viability of a neonate. Next slide, please. In terms of whether the trial occurs in the post-marketing setting with an approved product or in the pre-marketing setting with a new drug that is being developed, all requirements of Subpart B have to be met. Risk assessment and benefit considerations may vary depending on the setting. So for example, the amount of data available to inform safety, efficacy, and dosing, the gestational age at which the participant is being enrolled into the trial and whether it is beyond the period of organogenesis, the seriousness of the disease, and the availability of treatment options. Next slide, please. Trials in the post-marketing setting may be easier to conduct as you have more data to work with. Considerations include the following. Have the regulatory requirements been met? Opportunistic pharmacokinetic study designs can be used to inform correct dosing in pregnancy. These are studies where pregnant people who are taking a drug as part of routine clinical care are enrolled into a study where they provide blood samples. These types of studies are considered minimal risk. Intervention trials may be appropriate if the benefit risk profile is favorable. Next slide, please. Now, if we move on to trials in the pre-marketing setting, these, of course, are more challenging due to limited safety, efficacy, and dosing information. The first scenario involves study participants who become pregnant during a trial. Considerations to allow continued participation in PK data collection include the following. Are the non-clinical data adequate to support lack of risk? do the benefits of continued treatment during the trial outweigh the risks? So the risks of exposure to the fetus, the risks of discontinuing the drug and destabilizing the medical condition, and the risk of switching to another drug or drugs with potential fetal exposure to an additional drug. So in this scenario, unblinding is generally recommended so that benefit risk considerations and discussions can occur. And then the participant would then be reconsented as a pregnant participant to allow continuation in the trial. Next slide, please. So the second scenario involves including pregnant people in the development plan. And of course, this is much more challenging. Considerations include when to include in terms of the overall clinical drug development program, the benefit risk considerations of the drug that's being developed. So for example, the current pandemic provides a good opportunity to include pregnant people in COVID-19 treatment and vaccine trials. If non-clinical study results are not available, once they do become available, pregnant people could then be integrated into the trials at that point. Next slide, please. So safety monitoring of trial participants would involve consideration for increased safety monitoring, cord blood collection at the time of delivery, collection of pregnancy outcome data, and then follow up on the infant for at least a year in a pregnancy registry or other type of observational study. Next slide, please. The eligibility guidance was finalized in November. It references the clinical trials and pregnant women guidance and discusses considerations for women who get pregnant in a trial to allow them to continue. Next slide, please. In terms of COVID-19, FDA has published 2 guidances that encourage the enrollment of pregnant and lactating individuals in phase 3 clinical trials, if appropriate, and the early conduct of developmental and reproductive toxicology studies. Next slide, please. So in summary, FDA is committed to advancing clinical trials in pregnant and lactating people. Regulatory advances are occurring, which is a testament to stakeholders' commitment and efforts in this area. There is growing consensus on the need to include pregnant and breastfeeding people in clinical research and to integrate them into the research agenda rather than defaulting to automatic exclusion. There are many challenges that exist, and these challenges are discussed in the PregLac reports and will be discussed as part of this meeting. But the time is right, and as we wait for HHS to respond to the PregLac implementation report, stakeholders need to come together to collaborate to continue to move forward. Next slide, please. Thank you for your attention.
Thank you so much, Layla. That was a great presentation, and thank you so much at the end for starting to talk about PregLac, which is a nice transition for us, as our next speaker is Aaron Powlik. So Aaron, you're up.
Great, thank you, Susie. Can we have the next slide? All right, thank you. So I'm Aaron Powlik. I'm Chief of the Obstetric and Pediatric Pharmacology and Therapeutics branch at the Eunice Kennedy Shriver National Institute of Child Health and Human Development. So I'm going to give a brief update on the PregLac task force and specifically its implementation and what the NICHD is already doing to advance these. And Dr. Bianchi sends her regrets. With all the COVID rescheduling, our NICHD council is occurring on these same 2 days. So she really wishes she could be here in person to give this talk. So I'll be giving this update in her stead. Next slide, please. So what this slide shows is sort of highlights one of the major problems. What this analysis was a look at clinicaltrials.gov and the number of trials that included pregnant women. So only 2% of those trials in this analysis included pregnant women. And clearly this indicates pregnant women are amongst an underrepresented group in research. The paper by Dr. Spong and Bianchi really highlights the fact that if we want to improve public health, we're going to need increased research inclusion of these groups so that we can increase public health for all. Next slide, please. This slide highlights some of the statistics around pregnancy. About 6.5 million women become pregnant every year. Over 90% are taking at least one medication, and 70% are using at least one prescription medication. So it's very high prescription use. We don't really necessarily understand what these medications, how these medications may function differently in pregnant people. 500,000 women have difficulty producing milk, and this highlights the importance of this clinical need that's very unmet. There are really no therapeutics in humans to enhance milk production. A lot of the reason we're in this situation is concerns over liability by companies that may conduct trials, inclusion in labels, as well as other institutions and groups that are not currently including pregnant people in their studies. Scientifically, and this certainly underlies a lot of the liability concerns, is the complexity of pregnancy. The fetus and the placenta change over time with gestation. It's not something that's just a static organs and developing body to consider. The timing of drug exposure may therefore be very critical in terms of what adverse effect it could happen or potentially a therapeutic effect for either the mother or the child. Physiological changes during pregnancy also affect pharmacokinetics and pharmacodynamics, what the body does to the drug and what the drug may be doing to the body. So this is something that is not well studied and can certainly influence fetal development and effects as well. External factors such as xenobiotics in the environment, toxins, et cetera, may be influencing drug disposition. Obesity, which alters the environment for the fetus, may also be a factor that interacts with drugs. And certainly there are other coexisting conditions or chronic conditions or acute conditions that may require medication treatment, which can then alter the effects on other medications and have overall effects on pregnancy. And also in lactation, many lactating individuals have to make the choice between what they perceive as the benefits of breastfeeding versus the perceived risks of taking a medication. And this is really a decision that's made in the absence of a lot of data and limited very much by the limit of assays for assessing medication levels in breast milk. Next slide, please. So as been mentioned before, in the 21st Century Cures Act, Congress mandated the task force, which became the PregLac Task Force, which was led by NICHD. And this really focused on, identifying and addressing gaps in knowledge and research for safe and effective therapies for pregnant and lactating people, including the development of new therapies. And I think that's an important aspect of this. Next slide. At the same time as the PregLac Task Force was getting up and running, NICHD had initiated its strategic planning process. The overall goals for this were identify where the NICHD should take the lead, what are the top priorities we really need to have ownership for, identify where the NICHD can partner and collaborate in various areas, and what are sort of the key investments we can make for future research, training, and infrastructure. There were several core principles that led the development of the strategic plan, transparency and stakeholder participation being two of them, and really going for evidence and data in terms of how we'd make our decision. This strategic plan was launched in 2020. It's a 5-year plan, and the link is below if you want to take a look at that. Next slide. I want to highlight one of the 5 scientific themes that came out of that strategic plan, and that is advancing safe and effective therapeutics and devices for pregnant and lactating women, children, and people with disabilities. Certainly not going to read all this, but I just do want to highlight the fact that baked into the strategic plan for the NICHD is advancing safe and effective therapeutics for pregnant and lactating people. Next slide. So just briefly, PregLac report in terms of recommendations was submitted to the Secretary of HHS in 2018. There were 15 total recommendations. We were then asked to develop an implementation plan for these recommendations. Next slide. The task force organized 4 working groups to address the implementation planning for these recommendations, one on research and training, one on regulatory, communication, and discovery. Next slide, please. So this implementation plan was sent August of last year to the secretary at HHS. We haven't heard back from the secretary. Eventually, this will go forward to Congress for consideration as well. All of the information on the PregLac task force can be found at the NICHD website. Next slide. So I want to, next couple slides, summarize some of the implementation suggestions and where they may likely fall to the NIH and likely fall elsewhere. First off is leveraging or expanding existing federal programs or networks. This may be expanding networks that don't include pregnant and lactating individuals, or taking those that do work with pregnant and lactating women and expanding them. We may need to establish a prioritization process for studying therapeutics used during pregnancy and lactation. This would be very similar to what we do for pediatrics through the Best Pharmaceuticals for Children Act, where we conduct labeling changes for pediatrics. Development of new research tools is a very high priority for the NIH. For example, developing preclinical models such as placental transfer, which we are currently collaborating with NCAS at NIH on that. We definitely need to expand our training in obstetric and lactation pharmacology, and also consider alternative trial designs, for example, Bayesian adaptive trials or pragmatic trials that are embedded in healthcare systems, and to work with other partners such as the EU Conception Program. Next slide. Some recommendations that are likely not for the NIH, but for others. For example, if there's a requirement from Congress for industry to submit study plans, the FDA will be heavily involved in that, and certainly thinking about that is one part of this meeting. Addressing ethical considerations, liability, research incentives, the Office of Human Research Protection at HHS, that falls squarely within their wheelhouse. Fostering education and awareness is going to involve a lot of stakeholders. There's a lot of different organizations and levels we need to conduct education at. Public-private partnerships are going to be critical, especially in the therapeutic space. Liability concerns, the National Academies for Science, Engineering, and Medicine have addressed this for vaccines, including recently for COVID-19, and they may be an organization which could pick up a lot of the evaluation of liability. And certainly some of these steps will require authorization by Congress. Next slide. So I want to highlight a few of the things that the NICHD and the NIH have already taken to implement some of the recommendations from the PregLac plan. First off, we've created several new NIH reporting categories that report on how much money is being spent on various grants. The link is here for that. Surprisingly, or perhaps not surprisingly, these terms did not exist before the PregLac task force was formed. Now we'll have a better ability to historically track and see how we are making changes for future funding for pregnancy and lactation. Additionally, I want to highlight the Pregsource Resource. This is a crowdsourced platform where pregnant individuals can sign up and share a variety of their experiences with pregnancy. We've now added a medication and supplement tracker to this so that we can begin to collect volunteer data on this. Next slide. One initiative that the NICHD is moving forward with is the Maternal and Pediatric Precision and Therapeutics Hub. This is a new initiative that functions to serve as a service center and science catalyst to provide knowledge and expertise to the scientific community. It will serve as a platform for innovative and multidisciplinary research and synergize with other resources and networks, as shown here on the panel on the right. We really do intend this to be a hub that works with a lot of different spokes to accelerate maternal and pediatric therapeutics. And I will add that the pediatric portion is promoted by the BPCA. Next slide. Lastly, I do want to highlight a nascent public-private partnership for the qualification of biomarkers for early risk detection of preeclampsia. This is a project the NICHD is partnering with under the development in the Metabolic Disorder Steering Committee of the FNIH Biomarker Consortium. Next slide. Just lastly, to highlight the foundation for the NIH, this was an nonprofit organization created by Congress back in the 90s with the mission to lead alliances and public-private partnerships that support the mission of the NIH. So thank you very much. Next slide.
Thank you so much, Erin. That was a great talk. Thank you for introducing us to talking about all of the recommendations and the implementation of PregLac and imprint and the innovative multidisciplinary research. And that leads very nicely into our next talk by Jean Sheffield, who's going to talk to us about the urgent and key areas of unmet needs for therapeutic development. Jean, you're up.
Thank you so much. And thank you all, everybody, for listening. I think this is probably the easiest talk and discussion over the next 2 days because I'm essentially preaching to the choir. Everybody in the audience, all 547 participants that I see are all very interested in the topic. And so my job, which is supposed to convince you all that this is important, will be quite easy. I do want to thank Erin as somebody that was sitting at the table for both PregLac task forces. It is very nice to see that it's not just decisions being made in a closed room with a bunch of people sitting around a table, but there are actually things that are resulting from it already. It's absolutely amazing. Next slide, please. I do have no disclosures by the way. Next slide. So essentially, I'm just a clinician. So I am I'm on here to give you the clinical perspective. So I wanted to just mention the patient comes to you or comes to every encounter with a clinician with the assumption that we, as the clinician, will be able to accurately diagnose and treat whatever medical issue they may have. They're also going to assume that the physician is able to perform that risk-benefit decision behind the scenes about everything that you suggest they take and give the patient or give them the best answer. We always see that, you know, on the commercials and all that. Discuss with your physician before starting or taking or whatever. So our job as clinical care providers is to be able to have that discussion with patients. And don't forget, in the pregnancy or in the pregnant patient, we're not just doing that risk-benefit thought process with us and the patient, but there is also the baby, either 1 or 2 or 3 babies in there at the same time. So we have to kind of have that risk-benefit thought for both the patient and her pregnancy. Next slide, please. So I think Erin, I think several people have already mentioned this. We have about 6 million patients in the United States that become pregnant every year with 4 million live births, give or take. It always varies a little bit, but give or take. Next slide, please. Probably a little bit lower with the COVID stuff going on. And again, as also was mentioned a little bit by both my presenters before, at least 70 to 80% of people take at least one medication during the first pregnancy or during the first trimester, and about 90% will take a medication during the pregnancy. 70 to 80% of those prescription medications in the first trimester are often associated with just the nausea, vomiting. And interestingly, patients will average up to about 3 medications per pregnancy and about 4 medications if you start looking at pregnant and lactating women. Unfortunately, about 98% of these medications have data insufficient to determine teratogenic risk, and most of the dosing studies don't include pregnant women. So, you know, the first slide talked about we as clinical care providers have to have that risk-benefit discussion. This slide kind of, you know, hammers it home that we don't have data to make those decisions or those assessments. Next slide, please. So I wanted to just mention a couple of different things on pregnancy and therapeutic agents. It's not just the prenatal vitamins. We also have to look at the nausea, the antiemetics for the nausea, vomiting, the analgesias. I mean, pregnant women for that 10-month time period, they get headaches, they get back pain, they get hip pain as they go through the pregnancy. So it's good to have information about basic things like analgesia. Their allergies still act up. So it'd be helpful if we had information about allergy medications. Then you start throwing in the medical complications. We have a very large percentage of women now that have delayed childbearing, and they're coming to us, you know, after the age of 35 or after the age of 40 with higher rates of diabetes, hypertension, asthma, heart disease, seizures. So all of these different medical comorbidities oftentimes require medication, of which we know very little about in pregnancy. Next slide, please. So we also have different things such as obstetric complications requiring medications. Our patients have preterm labor, preterm rupture of membranes, they may hemorrhage, They may develop preeclampsia. All of these things we end up treating with different medical therapeutics, again, with limited information on how to counsel the patients. And then, fortunately, again, my other colleagues mentioned the lactation considerations also. This is not just pregnancy, but this is also pregnancy and lactation. And the majority of medications don't have a lot of information about breastfeeding or the effect on breastfeeding or how much of the medication passes through the breast milk to the infant. Next slide, please. Then I wanted to mention severe maternal morbidity, again, preaching to the choir, but figured I'd just at least mention it very briefly. These are any physical and psychological conditions that result from or aggravated by pregnancy. It affects more than 50,000 women in the United States annually. And again, because of the aging population and the increasing obesity problem in the United States, this SMM is increasing. So, we're seeing more and more of it, requiring more medical therapeutics. Next slide, please. I just wanted to mention a couple of these that we see quite a bit, and you can see this is hypertensive disorders. This is the data that's available right now at the CDC through 2014. Hypertensive disorders are going up. Postpartum hemorrhage is going up with a resultant need for medications. Pulmonary embolus is going up, even though DVTs have gone down a little bit. Next slide, please. And this is just trends in pregnancy-related mortality in the United States. Pregnancy-related mortality is any death of a woman while she's pregnant or within 1 year of the end of pregnancy that is either a cause related to or aggravated by the pregnancy or management. And I wanted to put some of the reasons why why somebody has a pregnancy-related death. And you can see in this slide, cardiovascular disease, infection, sepsis, cardiomyopathy, hemorrhage, pulmonary embolisms, hypertensive disorders. All of these things require therapeutics. Next slide, please. I did want to mention, we have been very, very good in the last 10, 15 years about starting to focus on maternal mortality and looking at where we stand in the United States versus the rest of the world, and truly starting to address at multiple different levels, from the individual provider's office up through our societies, up through the CDC and the FDA. Again, pretty much everybody on this call is involved now in this initiative to try to decrease maternal morbidity and mortality. And this is just one thing I always want to put in there as kind of a plug for SMFM. SMFM is actually looking now at state level maternal mortality facts and figures, kind of giving information about do they have review committees, et cetera. So we're starting to be able to delve down a little bit more with the help of a lot of our societies. Next slide, please. So basic call to action. This is what Aaron was talking about, the 21st Century Cures Act and PregLac. I am beyond thrilled that we are starting to see a true mobilization of multiple different aspects towards decreasing maternal morbidity and mortality and trying to address the problem of lack of information in pregnant women. Next slide, please. So let me just go back to the bedside. I have like 2 minutes left. So what we as clinical care providers need to know about every medication out there, everything from Claritin, Allegra, Tylenol, on up to our antihypertensive medications or some of the cardiac medications we have to use for a heart transplant patient in pregnancy, et cetera. We need to know something about every medication that is out there. We need to know safety and toxicity data, including teratogenic risks. We need to know about dosing information. Again, do you need to dose it differently in the first trimester compared to the third trimester? Or do we need to adjust dosing after they deliver? And what are the considerations if they are or are not breastfeeding? We need to know all of those things. We also need to know about what the effect of BMI is on these different medication dosage or different dosage issues. Next slide, please. So I just want to briefly mention, and we're going to have several discussions about this, the ethical principles of pregnant women and biomedical research. Pregnant women deserve an evidence base for the prevention and treatment of their illness equal to others as a matter of justice. And this was a statement that I took directly out of the Ethics Working Group on Zika research in pregnancy in 2017. And I think it addresses the issue that we're discussing over the next couple of days perfectly. PregLac also discusses the inclusion or a scientific justification for exclusion of pregnant women in drug-related studies. And I think also, and I know this is also being discussed, I believe, tomorrow, you got to remember pregnant women, you know, clinical care providers have a tendency to be very paternalistic or very protective of their pregnant women and try to avoid, you know, putting them in studies and such. But on the flip side, you take a pregnant woman and ask her if she'd be interested in being involved in studies. Hey, can we draw an extra tube of blood here and there, whatever. The vast majority of them are absolutely going to say yes, not just for their potential benefit, but for the benefit of other pregnant women. So there is definitely an altruism aspect of research in pregnancy. Pregnant women are not reticent to be involved in research. So I wanted to put that out there again, though I know we're talking about it a little bit tomorrow also. It's important, and they recognize the importance. Next slide, please. And with that, I'm going to, I'll stop. I think I went 1 minute over, so I will stop, and I know we're going to address some questions. Thank you.
Jean, thank you so much for a fabulous talk. And I just want to thank all of the presenters in this first session for very informative presentations. And I would just ask that Jean, Aaron, and Layla all come back on camera, please, and unmute yourselves. Excellent. And then maybe we can go to that next slide, the discussion questions, please. So we're going to move into the moderated discussion portion of this session. And I recognize that we only have a couple of minutes, but we were really trying in this first session to lay the groundwork for the future discussions in the additional sessions. So I'm going to ask a couple of questions here up front. I know we have a few in the Q&A box. The one I do want folks to answer is, what are the gaps in information on the safety, efficacy, and dosing of drugs used in pregnancy? And what are the risks of not having that information? And so while you're thinking about that, I'll just read through these other questions that we may not be able to get to because we have a couple in the chat box. And then how do we determine the magnitude of benefit that this type of evidence would provide? The second question, what have we learned so far from the work undertaken by PregLac since its inception in 2016? And what are the regulatory barriers to research involving pregnant people? So maybe we'll take those questions off and we'll move to our opportunity to have our panelists discuss that first question about the gaps in information and what are the risks of not having that information and the magnitude of benefit that this information would provide.
So I'll jump in then. I can jump in just real briefly from a clinical standpoint. There are a lot of medications that I have to use in my pregnant women and pregnant women for management of their medical conditions. And pregnant women can be very reticent. They don't want to take a medication if it's going to harm the kid, harm their fetus, harm their baby. And so these pregnant patients, I need the information to be able to give it to them so we can make an informed decision about what is the benefit of that medication for the treatment of the maternal condition, and how do we optimize her dosing without compromising the fetal safety. And so we desperately need that information to be able to optimize the mom's health while keeping the baby healthy.
Thank you, Jean. I think, Aaron, you were up next.
Yeah, I would add, I think we also don't necessarily understand what a lot of the gaps in that knowledge is. That's one of the goals of the Imprint Hub. One of the components is a knowledge resource and coordination center that will start to pull a lot of that information together. That's a little bit perhaps earlier than the actual clinical and labeling information. But I'd say overall, there's a lot we don't know, and there's a lot we don't even know that we don't know. So we need to start addressing this landscape more broadly. There's also some efforts underway that are going to be very important in terms of connecting mother to baby medical records so that we can start to look at long-term outcomes from drug exposure. And so building up the infrastructure so that we can more effectively monitor and pull this knowledge together moving forward is going to be really important for closing those gaps.
And Layla, did you want to weigh in there?
Sure. Yeah, I think that, I mean, we see the gaps because we work on labeling every day. And so, you know, why does every other section of prescription drug labeling have information, but when it comes to the pregnancy section, there's no data? There's no data to inform the safety, the use, or dosing. So I think that that is a challenge that we deal with every day with newly approved drugs. In terms of the risks of not having this information, so the risk gets pushed to the prescriber, right? So it's the prescriber that then has to have that conversation with the pregnant woman in terms of making those prescribing decisions in the absence of data. So that's where the risk gets pushed out to that setting.
Well, thank you so much. I want to thank Layla, Aaron, and Jean for fabulous talks. We're going to try to keep on time here. I will just give Aaron, for future conversations, a heads up that one of the questions was about the mandate for public-private partnerships. And then one of the other questions had to do with the data that Jean mentioned associated with the non-clinical data that we have associated with products, which is a lovely segue into our next session. So I want to thank everyone for a fabulous discussion that really kicked off, I think, what's going to be a really informative meeting. And now we're going to move to session 2, which is going to be moderated by Daniel Mink of the FDA. And so with that, I will turn it over to you, Dan.
Thank you, Suze. Good afternoon, everyone. As you heard, my name is Dan Mink, and I am from the FDA. And I get to be the moderator of the session 2, which talks a little bit about the non-clinical safety assessments that are performed to support the enrollment of women in clinical trials. We have 2 speakers in this session today that will be giving a joint presentation. We have Dr. Kim Hatfield, who is a team leader in the Division of Urology, Obstetrics, and Gynecology at the FDA. And she'll be presenting material pertaining to the conduct of the non-clinical studies used to support women in, individuals in pregnancy. And we have Dr. Leslie McKinney, who is a pharmacologist in the Division of Rare Diseases, Pediatrics, Urologic, and Reproductive Medicine. And she'll be talking more about what do we do with these data once they are generated. Both Kim and Leslie have extensive experience providing consultation and writing guidances for the non-clinical review, focusing on reproductive toxicity. So the presentations today will cover the characteristics of a non-clinical testing program to support enrollment of individuals who may be pregnant or want to become pregnant, potentially on clinical trials. And they'll be talking about how such a lack of such data makes it difficult to predict any clinical safety or risk for a pregnant individual in particular. And they will also address how the data will be used in the risk-benefit assessment. Main takeaways that we'd like you to come away from this presentation are an idea and understanding what the non-clinical data can tell us to inform on risk for the population of interest, and how in particular good detailed non-clinical studies can support enrollment of pregnant individuals in clinical trial. I also hope to get the point across that, the studies that are typically performed, they're guideline studies. That means deviations from those guidelines are fine if you can justify it. And in some cases, how it may be beneficial to do a little bit beyond the typical guideline studies. And thirdly, I'd like to ensure that people get the idea that the FDA integrates all the available data to assess and communicate risk in the form of the label ultimately. However, as you heard from Layla earlier today, that there's not necessarily all this information that is available prior to the marketing of a drug. And so we want to relay the information that we typically do obtain that helps us make a decision about whether or not it is safe to go into pregnant individuals with the drug. And before I turn things over to Kim and Leslie, I just want to remind everyone to feel free to submit questions using the Q&A function on the bottom of the screen. With that, I would like to turn it over to Kim and Leslie.
Thank you. Thank you so much, Dan. Next slide, please. I just want to say again, good afternoon, and give my thanks to the organizers and my colleagues for creating such an excellent workshop today and giving both Leslie and I the opportunity to talk about the non-clinical safety assessment that is used to support clinical trials during pregnancy and inform us on drug exposure during the prenatal period. As mentioned, Leslie and I are tag teaming this presentation today, and we are both pharmacology and toxicology reviewers at the FDA, and we support the clinical division of urology, obstetrics, and gynecology. Next slide, please. So our objectives today are to first explain what types of animal studies are conducted to predict the safety of a drug substance to a developing human during the prenatal period, and then also describe the data that support the pregnancy section of the product label. Finally, we'll discuss what data gaps exist even after the standard reproductive and developmental toxicology studies are completed, and what kinds of non-clinical studies might be necessary and specially conducted to support clinical trials in pregnant individuals. Next slide, please. So the first question that many of you may be asking, why do we even need to conduct non-clinical toxicity studies? Well, the first answer is pretty obvious in that they are animal studies to evaluate drug toxicity are mandated. And this is documented in the 1962 Kefauver-Harris amendments to the Food, Drug, and Cosmetic Act. And these were written in response to the thalidomide tragedies of the late 1950s and early 1960s. And these amendments document that there needs to be a demonstration of both the safety and the efficacy of marketed drugs. And our Code of Federal Regulations, or 21 CFR, also documents that animal toxicology studies are necessary to support first-in-human clinical trials. Now, over a number of years, we have developed regulations and guidance for both how and when to conduct non-clinical studies. We've done that through international guidance in conjunction with other international regulatory agencies from Japan or the EMA, and also in concert with the International Council for Harmonization, or ICH. We also have a number of FDA guidances, which can be found at the link noted here, and that can also guide the sponsors as to how and when to conduct non-clinical studies. Now, we're going to be focusing today on the reproductive and developmental toxicity studies that mainly inform use of a drug during a pregnancy. But these studies are just one part of a number of studies that form the total non-clinical assessment of a drug product. Next slide, please. So what do reproductive and developmental toxicity studies measure? First of all, reproductive toxicity and developmental toxicity are two very separate entities. Reproductive toxicity focuses on evaluating the structural and functional alterations that affect reproductive competence in sexually mature males and females. And this involves evaluating drug exposure and how it influences male and female fertility, parturition or labor and delivery, and lactation. So reproductive toxicity mainly focuses on the pregnancy. Developmental toxicity, however, focuses on evaluating the adverse effects on the developing organism, or as some may know it as teratogenicity. And this evaluates the effects of drug exposure on mortality of the offspring, any alterations to growth in the offspring, structural abnormalities, and functional impairment. So the developmental toxicity portion focuses on such as that, the development of the offspring in particular. Next slide, please. So I'm going to be covering objective 1 today of our talk, and that is to focus on what kinds of animal studies are conducted to predict the safety of a drug substance to a developing human during the prenatal period. And then what are the characteristics of a comprehensive non-clinical program? Next slide. So how do we evaluate reproductive and developmental toxicity? Well, as I had mentioned before, these studies are part of a number of non-clinical studies that we use to evaluate risk to human exposure. And the first way that we can touch on looking at any reproductive effects of a drug are to conduct repeat dose toxicology studies. These studies are conducted in non-pregnant animals and typically involve dosing with a placebo and then a low, mid, and high dose of drug, usually during daily administration over a specified duration of time. And then animals are terminated to evaluate drug exposure and how it affects different biological processes, such as body weight, hematology, gross pathology, and histopathology are just a few. But for the reproductive evaluation, we can use these studies to look at the histopathology of the reproductive organs and to see if there's any effects both on a cellular and microscopic level. Also, we can evaluate the estrus and menstrual cycle if these parameters are included in the repeat dose study. And if we see any extended time at any one stage of the cycle, we can kind of infer that there may be some hormonal imbalance occurring or some disruption of feedback mechanisms that could influence reproduction. Finally, we have specific reproductive and developmental toxicology study designs, and these include the fertility and early embryonic development study, the embryo fetal development study, and the pre and postnatal development study. And I'm going to go over these in a little bit more detail in the following slides. Next slide, please. So the schematic presented here shows that the reproductive and developmental cycle is defined by 4 biological phases represented by the different colors, and it's separated into 3 dosing segments represented by the periods between the hashed lines. So if we start on the right, we have mating, and that is representative of the adult male and female who are sexually mature. And then we can look at gestation of the offspring following in a clockwise direction, gestation of the offspring, pre-weaning, and then maturation of the offspring. The 3 dosing segments are separated from the right-hand side. We see between weaning and implantation of the embryo in the uterus. The second dosing segment is implantation to parturition. And then the third is examining from parturition to weaning. And we have different toxicity studies to evaluate each of these segments. The first on the right is the fertility and early embryonic development study. And this examines drug exposure to a sexually mature adult female and male and evaluates any drug effects on fertility, ability to mate, and whether or not an embryo can be implanted in the uterus. The focus of our talk today is going to primarily dwell on the embryo fetal development and the pre and postnatal development studies, since this involves dosing during pregnancy. So the embryo fetal development study at the bottom covers the time period of dosing from implantation up until the point of delivery. And then on the left, we see the pre and postnatal development study that covers the period from implantation of the embryo in the uterus through delivery and up until weaning, during lactation through weaning. Next slide, please. So the first study I'm going to focus on is the embryo fetal development study, or EFD. And these studies are really important for us to evaluate drug exposure for drugs that could be used during pregnancy. Jean had mentioned that there are a number of drugs that women take during pregnancy, not for a pregnancy condition, but just to treat a multitude of different medical needs, such as diabetes or any plethora of indications. So these studies are very important to evaluate what could happen in an animal after taking those types of drugs. So these studies are evaluated in 2 species, a rodent, usually a rat, and a non-rodent, usually a rabbit. And dosing occurs from implantation through organogenesis up until closure of the hard palate. So the schematic shown here, if we start on the left, at the time of mating, that is represented in the rat and the rabbit as gestational day 0, or GD0. Dosing begins at implantation, which is generally day 6 or day 7, and continues daily with dosing of the mother during organogenesis up until gestational day 17 or day 19, depending on the species. At this point, dosing is stopped. The animals maintain their pregnancy for a little while longer, and then just prior to delivery, the animals are terminated and C-sectioned, where both the mother and the offspring are terminated in order to evaluate different toxicity endpoints. These studies involve at least 16 pregnant females per dose group, and with an average litter size of 6 in the rabbit up to 12 in a rat, we really get a large number of offspring in order to evaluate any findings that we see. And this is necessary for a level of statistical power, but also to be assured that any findings are increased above the natural background of pregnancy. Next slide, please. The EFD study evaluates a number of different endpoints. The first are maternal health endpoints, where we look at the drug effects on body weight changes of the mom and evaluation of any general toxicity to organ systems relative to what has previously been shown in studies involving non-pregnant females. We then look at embryo fetal development and other pregnancy parameters, such as drug effects on gravid uterine weight, the number of fetuses per litter, pre and post-implantation embryo fetal loss, and then also gross evaluation of the placenta. So this is where we look at what happens during the pregnancy. And then finally, we look at the fetal health endpoints and how did drug exposure alter growth of the offspring, alter their body weight, and we also look at any gross evaluation of external, visceral, and skeletal development that may have occurred due to drug exposure. Next slide, please. Next, we have the pre and postnatal development study, or Ppnd. And this study is also important for drugs that could be used during pregnancy or for those that are administered during lactation. These studies are evaluated in 1 species, usually a rat, and dosing occurs from implantation through delivery to the end of lactation. In the schematic shown here, we see on the left the representation of mating of the F0 generation, which is the adult male and female. Dosing of the moms begins at implantation or gestational day 6, continues daily through gestation. The moms are allowed to deliver, and then the moms continue to be dosed daily through lactation up until postnatal day 21, when the offspring are weaned. At this point, the moms and the offspring are terminated, and 1 male and 1 female offspring per litter is selected for rearing to adulthood and mating to evaluate reproductive competence. And as with the EFD study, there are at least 16 pregnant females per dose group to allow for a good amount of statistical power to evaluate the findings. Next slide, please. The endpoints assessed in the Ppnd study are slightly different from the EFD in that it focuses more on the development of the offspring. The first things we look at are the maternal health and maintenance of pregnancy during treatment and drug effects on body weight changes, duration of pregnancy, any pregnancy or whole litter loss, and any live or dead offspring at birth. We then look at offspring that were exposed in utero and via lactation, and look for any effects on body weight, survival and growth, sexual maturation, fertility and reproductive capacity, any changes to physical development, and then changes to sensory function and reflexes or behavior learning and memory. And then finally, the offspring of, or the F2 generation that were not exposed, but the parents were, so they were exposed, the parents were exposed in utero and during lactation, were allowed to reproduce. We examine their generation by external exam and survival to postnatal day 4 to 7, just to make sure that the exposed animals were available to reproduce. And this also looks at kind of a grandfather effect of the drug on this, I guess, the second generation. Next slide, please. So now I want to segue into the timing of non-clinical studies that occur during pharmaceutical development and clinical trial conduct as we progress from the investigational new drug phase, or the IND, up until the NDA. So the schematic here represents the time period from drug discovery through IND clinical trial phases 1, 2, and 3, and then ultimately NDA submission. IND phase 1 represents the first in human clinical trials. And at that point, prior to conducting these studies, we need to have certain non-clinical information. And that includes information on the mechanism of action of the drug, any receptor and organ targets for the indication. We also need to know how the drug works in the body. How is it absorbed, distributed, the metabolism of the drug, how is it excreted? How long does the drug stay in the circulation? Does it break down to toxic metabolites? We also need to know the general toxicity in non-pregnant animals, both males and females. What are the target organs? What are safe doses? And does it affect reproductive organs? And then finally, we need to know the genotoxicity and whether or not the drug is a mutagen or if it can damage DNA. And all of this information is collected by the sponsor and submitted to support their IND phase 1 clinical trial. Next slide, please. So during the IND, if a sponsor is conducting a traditional clinical trial in a general indication, these clinical trials are going to enroll males, postmenopausal individuals, and/or individuals of childbearing potential, with the caveat of pregnancy testing and contraceptive use written into the protocols. During the clinical development for this type of drug program, the general toxicity studies with reproductive organ assessments are conducted during early drug discovery and during the phase 1 clinical trials. The fertility and embryo fetal development studies are completed during and between phases 2 and 3 clinical trials. And then the Ppnd studies are generally conducted concurrently with phase 3 and are completed and submitted at the time of NDA submission. Next slide, please. However, there is a big difference if the clinical trial is going to be investigating a pregnancy-specific indication, such as preterm labor or hyperemesis gravidarium, or if the clinical trial is going to be enrolling pregnant individuals, such as a drug to treat epilepsy, and they just want to find out more about how that drug is going to affect a pregnant population. So these trials specifically enroll actively pregnant individuals. The caveat with these types of studies is that all reproductive studies, the fertility, the EFD, the Ppnd, along with the general toxicity and genotoxicity, must be complete before any pregnant individuals are enrolled in a clinical trial. So that really puts a higher burden on the sponsor to get those studies completed prior to conducting a clinical trial in a pregnant population. Generally, for these types of studies, phase 1 includes non-pregnant subjects because their sponsor is trying to get an idea of the pharmacokinetics and any dose range finding. But then at the time of phase 2, those generally enroll pregnant subjects. So all of the studies need to be completed prior to that point of time. Next slide, please. So I've explained all of these specific studies, but how do we actually use the data that's generated for them and extrapolate the animal findings to humans? So we've conducted the repeat dose, the EFD, and the Ppnd studies. And those studies will generally yield a value for a dose that produces no toxicity in animals. And we define that dose as the NOEL, or the no adverse effect level. The NOEL is then compared to the recommended human dose and is divided, so the NOEL is divided by the therapeutic dose in humans, and that generates what's called a multiple of exposure, or also called a safety margin. And the larger the multiple of exposure, the safer or the less risky the drug. So for example, a NOEL could be found in a rat at 20 milligrams, and the therapeutic dose in humans is 2 milligrams. So that would generate a multiple of exposure of 10. And that would indicate that at a dose 10 times the therapeutic dose, there was still no toxicity in animals. So that gives you a nice safety margin to estimate the level of risk that you're going to be looking at with drug exposure in that population. So the multiple of exposure and the totality of safety information about the drug are then used to guide dosing decisions and communicate risk. Next slide, please. So the animal data also informs human risk when there is very limited or absolutely no human data available. If the animal studies are negative and show no safety findings whatsoever, that animal data can really support further clinical testing without too much risk. However, if there are positive findings and we see it like a major toxicity, the animal data can still help us define a safe human dose range. And that comes into play when we look at the multiple of exposure. If there is a large multiple of exposure where at a point where we see a positive finding, that indicates less risk. There may not be as much of a risk of this happening in the pregnant female or in a pregnant individual whatsoever. However, if the multiple of exposure is small, say the toxicity finding is only shown at 1 or 2 times the therapeutic dose, then we have more risk and we need to really evaluate that to make sure that we have a safe dosing range in the clinical population. There are limitations to animal data, and clinical trials could either be rejected or asked to have their protocol significantly modified if the animal data shows some sort of potential harm to the fetus that cannot be detected until birth. And that would be considered a non-monitorable toxicity, such as neurobehavioral development, that you're not going to see until postnatal day 21 or so in an animal. Also, if exposure to the fetus cannot be determined, then we have to really take a low and slow approach in evaluating that safe human dose range that could be used. And then finally, if the risk benefit is unfavorable, say the risk is just too high for a low benefit, then we have to reevaluate. But if human data does become available over time, it can contribute to the weight of evidence to assess safe use. So maybe you have a drug that's going to be used in preterm labor, but it's also indicated for something different, like use in asthma, for example, just hypothetically. There may be some pregnancy registry data from that indicated drug for asthma that could inform use of its use during preterm labor in terms of the dosing, in terms of what could be found in the clinical population. And that helps the weight of evidence approach. Next slide, please. So now I'm going to turn it over to Leslie for objectives 2 and 3. And Leslie's going to talk about how our non-clinical data can actually be used in the risk assessment and regulatory evaluation of our drugs. Take it over, Leslie.
Thank you, Kim. Now that everyone hopefully has a clear idea of what kinds of animal studies support clinical dosing, our next objective is to explain how the results of these reprotox studies are used to communicate risk once a drug has been approved. For that, we're going to look at a couple of labels and show you how the data from reprotox studies are incorporated into the pregnancy section of the product label. Then we will talk about how non-clinical data play a critical role in FDA's risk-benefit assessment for new drug development. Next slide. Instructions for how to write a label to guide drug use in pregnant women are spelled out in the Pregnancy and Lactation Labeling Rule, which was implemented for all new labels about 5 years ago. It replaces what used to be called pregnancy categories, which used letter designations A, B, C, D, X to communicate risk. These letter designations were sometimes misunderstood to imply a gradation of risk, which was not quite correct. So the letter designations were eliminated, and the pregnancy section, Section 8 of a drug label, must now contain a discussion of risk-benefit along with the supporting clinical and non-clinical data. Sections 8.1 and 8.2 cover women who are pregnant or lactating and must contain a risk summary for use, clinical considerations, and relevant human and animal data. Section 8.3 covers males and females of reproductive potential and contains information about whether patients should undergo pregnancy testing or use contraception while they are on the drug, and whether there's an effect on fertility. Next slide. Now, we all know from reading product labels that in general, human data are not available for drugs that might be taken during pregnancy, or any other drugs for that matter, and that animal data have to be used to inform the communication of risk to the patient. There are some exceptions to this, for example, in the case of estrogen products. Because there is so much human data accumulated over decades related to estrogen use, Section 8 of these product labels does not need animal data to inform risk. But for most drugs, animal studies are in the labels, and so next we want to go through how you can use that data to understand the risk profile for use in pregnant women. Next slide. To illustrate these points, we chose 2 drugs with different risk profiles for pregnant individuals. First, we will look at Prevacid, which would be taken episodically during pregnancy for a non-life-threatening indication, namely acid reflux, and Humalog, which is a fast-acting human insulin analog, which would be taken during pregnancy for a life-threatening indication, namely diabetes. Now, just as an aside, we note that neither of these drugs is for a gynecological indication, so our division was not directly involved in approving these drugs. They were approved by other divisions within the FDA. Their labels are presented here for instructional purposes only. Next slide. This slide shows text from Section 8 for the Prevacid label. For clarity, the text for this label and the next one has been edited and condensed to make the slide easier to read. The first part of Section 8 is the risk summary, which gives the high-level overview of risk based on human studies first, if they are available, and animal studies second. So for Prevacid, data from observational studies in humans are negative. They have not shown evidence for adverse pregnancy outcomes. The animal studies, however, do show some adverse findings, namely that in the rat Ppnd study, there was some effect on growth. However, this was only observed at 6.4 times the maximum recommended human dose. Based on that finding, the risk statement advised there may be a potential risk to a fetus. So here you have a case where there's no safety signal in humans, but there is a finding in animals. Next slide. The second part of section 8.1 contains additional data that further explain the animal findings. The first paragraph gives the results of the EFD study, which showed no adverse effects in rat and rabbit at high multiples of exposure, 40 and 16 times the maximum recommended human dose, respectively. The second paragraph gives the results of the Ppnd study, which has some findings related to both the pregnant females and the pups. The Ppnd study was conducted over a good dose range, 0.7 to 6.4 times the maximum recommended human dose, and contained some non-standard endpoints on bone development. What you should notice is that the high-dose females did not gain weight as they should have during pregnancy, and that the body weight of the pups was also reduced. Reduced growth of the pups was then reflected in the bone parameters. So the conclusion is that at a dose 6.4 times the maximum human dose, there was some maternal toxicity that had some adverse effects on the pups. Within this context, it is probably safe to conclude that this would not happen in humans, and therefore the drug has a low risk for use. This can be balanced against the benefit of reducing the discomfort of acid reflux during pregnancy. Next slide. Now for Humalog, a fast-acting insulin. The risk statement starts off with a statement about what is known about the risks of taking the drug. There are no negative findings reported for the mother or the fetus, but it is also clear that the human data are not very robust. Next, the risk summary states clearly that there are risks to the mother and the fetus of not treating the disease. So what do the animal data say? The EFD study in the rat showed fetal growth retardation at 3 times the human dose, but the EFD study in the rabbit showed no adverse effects. However, note that dosing in the rabbit did not reach the level of the human dose, but only a fraction of the human dose. So the animal data are equivocal, but not strongly negative. Next slide. The next section of the risk summary, clinical considerations, again emphasizes the risk associated with doing nothing. The animal data section gives more detail about the rat study, which was a combined fertility and EFD study. It showed that fetal growth retardation was the only finding, and then only at doses above the therapeutic dose. Everything else was negative. In the rabbit, there were no findings, but again, dosing did not reach a very high level. So the animal data are not completely clean and indicate some risk, but that has to be balanced against the risk of untreated diabetes. Next slide. So for the last section of this talk, we want to address what data gaps exist even after the standard Reproductive studies are completed. And then we want to address what other kinds of non-clinical studies might be necessary to support clinical trials in pregnant individuals, especially if there's a concern that the standard Reproductive studies may not give us enough information to do a full safety evaluation on a drug being considered for a clinical trial in pregnant women. Next slide. So what are some of the limitations of standard Reproductive studies? Well, there are the inherent limitations, namely the species differences. Rats and rabbits are not humans. And there are developmental differences. Although all mammalian species go through the same sequence of developmental stages, each species matures at a different rate. So for example, when a rat is newborn, it is actually comparable to a 3rd trimester human. Other limitations are due to how the studies are designed. For example, Reproductive studies are primarily observational. They assess only gross morphological abnormalities, only basic physiological functions, and only simple instinctually driven behaviors. Next slide. There are many parameters that are not typically assessed. For example, in neurodevelopment, sensory modalities are not assessed for subtle changes. Higher order learning and memory are not usually assessed. For immune system development, there's no systematic evaluation of immune competence. Now keep in mind that lab animals used in reproductive studies are raised in a very clean environment. They're not exposed to many pathogens. So it might be difficult to know whether a lab animal would respond adequately to all of the pathogens that they might encounter in the wild. Lastly, the endocrine system is not usually assessed. Onset of puberty and bone parameters are not usually measured, but puberty can be evaluated, but in a separate kind of study called the juvenile animal study that has a very different design. Next slide. Any number of additional tests can be conducted to evaluate risk if that is deemed necessary. For example, if you want to know more about fetal exposure, you can measure placental transfer. You may want to measure tissue concentrations of the drug or metabolite in the fetus. You may want to show effects on specific systems and find out whether certain organ systems are vulnerable. You may want to do developmental studies in the 3 areas I mentioned previously for brain and behavior, immune challenge, or endocrine function. We wanted to show an example of a label that includes results from this sort of extra testing. So we chose Lactating, trade name Cymbalta, whose label includes extra neurobehavioral evaluation of offspring. Next slide. Cymbalta is a serotonin norepinephrine reuptake inhibitor approved for several psychiatric and neurological indications. The risk summary has some human data and indicates no known human risk. Animal studies indicate no teratogenicity, that is, no findings during the period of organogenesis. But they do describe some developmental effects at 2 times the maximum recommended human dose, namely reduced growth and survival and pup behaviors consistent with increased reactivity. But what exactly is increased reactivity? Next slide. When you look at the additional details from the animal studies, you see in the second paragraph that the statement about increased reactivity was based on endpoints such as an increased startle response to noise and a decreased habituation of locomotor activity. Now, what does that mean, and how do you extrapolate that finding to humans? Well, that is where it gets hard. The label has to report any positive findings that occurred, but especially in behavioral studies, these types of results only indicate a potential risk to humans. They may lead to further study in animals on the mechanism of action of the drug, and they may indicate the need for long-term follow-up studies in humans. The bottom line is you look at the multiple of exposure and the severity of the findings, and you evaluate risk-benefit based on that. Next slide. So now I want to give you a little bit of insight into what goes into the decision-making process when a new clinical trial protocol lands on our desks at the FDA, and we have to determine whether the investigator should be allowed to proceed with the trial. A review team is assigned to review the protocol, which has members from each scientific discipline: chemists, clinical pharmacologists, toxicologists, and clinicians. The review team integrates all the information and results from both non-clinical and any available clinical studies. Then we spend a lot of time discussing how relevant the animal findings are to humans, and we especially focus on fetal risk. We ask a lot of questions. Can we estimate the fetal drug exposure? Do we know how the drug is absorbed, where it's distributed, how it's metabolized? Especially we want to know if the fetus will be exposed and how much compared to the mother. Of course, we consider how long and during which trimester the fetus might be exposed and whether the fetus can metabolize the drug. During development, the fetal liver is maturing, and metabolism may be very different at the end of gestation as opposed to the beginning. Of course, we want to know if the fetal brain can be exposed, and we want to know if there are any toxicities that could affect the fetus more than the mother. These questions sometimes require us to consult subject matter experts, of which there are many at the FDA, and if we need additional input and guidance. Next slide. So to walk you through what this process might be like, we designed a fictional case study to illustrate what might happen when a company wants to develop a drug for a pregnancy-specific indication. So let's pretend that small innovative drug company is developing a wonder drug for a very serious adult disease. During development, their animal studies revealed that wonder drug has toxicity. It has cardiac toxicity, but only at very high doses, 30 times the proposed therapeutic dose. So the company was allowed to go into phase 1 clinical trials. And during the phase 1, no toxicity was observed at the therapeutic dose. So they proceeded to phase 2, but Wonder Drug failed in phase 2 due to lack of efficacy. But the small innovative drug company doesn't give up on the drug. They continue to do research, and they found out that their drug might be effective for prevention of preterm labor. So they designed a phase 2 trial to enroll pregnant individuals who would be at high risk for preterm labor because they've already had one previous preterm delivery. They proposed to start dosing at 18 weeks of pregnancy and to continue throughout gestation at the therapeutic dose that they already showed from their previous phase 1 trial to be safe in adults. Next slide. So the question is, can small innovative drug companies show that the Wonder Drug is also safe for the fetus? Their previous animal work in the rat had shown that their drug crosses the placental barrier, which indicates that the fetus could be exposed. Their EFD studies in the rat and rabbit showed dose-dependent fetal loss, but that was at very high doses where a Wonder Drug caused maternal heart damage that may have contributed to the fetal loss. However, even at lower doses that didn't affect the pregnant females, there was still a low incidence of fetal loss, raising the question of whether their drug was affecting cardiac development. The good news was that the no effect level, or the NOAEL, for fetal loss was 10 times the therapeutic level. So the question is this margin enough to be sure of safety? Or put another way, is fetal loss a sensitive enough endpoint? Next slide. So how can the company proceed? At this juncture, communication with the FDA would be helpful to determine what kind of safety data are needed. Let's assume that small innovative drug company schedules a meeting with the FDA, and the FDA recommends that additional animal data should be obtained. But what kind? Well, realizing that the Ppnd study is yet to be done, the FDA might recommend that additional endpoints be added to explore whether there was cardiotoxicity in the fetal rats that might have been missed in the EFD study. For example, the company could do histopathology of fetal organs and try to find the cause of fetal loss. They would then be able to establish a no effect level, or NOAEL, for cardiotoxicity in the fetal animals. Once they get a dose dependence of these specialized endpoints, it would then be possible to set a safe dose in humans based on the multiples of exposure. Next slide. So you can see when it comes down to making a decision about a clinical trial, it all comes down to benefit versus risk. You have to assess how serious the indication is for the mother, for the fetus, whether the benefit is certain, whether the population is well defined. If the drug is toxic to the fetus, is it monitorable? Is it reversible? And lastly, if the mother's not treated, will her condition be a risk to her fetus? We hope that our talk has shown you that reproductive developmental toxicity studies in animals can provide essential information to the clinician to use to make a treatment decision and to inform the patient of risk and to hopefully enable clinical trials in pregnant women. Next slide. We're including a list of guidances for you to refer to about non-clinical testing. And lastly, I want to thank my non-clinical colleagues at the FDA for their help and input in developing this talk. Thank you very much for listening.
So thank you, Kim and Leslie, for your presentations. I thought they were very interesting. I would like now to move into the moderated discussion portion of the session, where we have a couple questions that we've already highlighted, and then I'm assuming we'll have some time for some of the questions that have been sent in at this time. So both Leslie and Kim can unmute themselves and turn their cameras on. We'll start with the first discussion question. And the first discussion question is in regards to what happens if a non-clinical safety signal is observed. Are clinical trials in pregnant people likely to be prohibited? Or what additional information could the FDA rely on to inform risk? What is needed for an optimal discussion of benefit versus risk that could lead to inclusion of pregnant individuals in trials when a safety signal is observed? So either Kim or Leslie, you want to take that one?
Is there a slide that shows the discussion questions? Just so that someone could see them on the page?
Can you answer that, Kaylee? Can you put that slide up?
Thank you so
Thank
much,
you.
Kaylee. So I can start off with this and Leslie can jump in. Just because we see a non-clinical safety signal doesn't mean that we're going to prohibit a clinical trial. I think it all depends on where, what we see, what kind of signal it is. It could be very strong or be very not as concerning. So, and it also depends on what dose it's seen. So we have to rely on that multiple of exposure to kind of figure out what our risk is. But it doesn't mean that the clinical trial is thrown away at that point. We could, I mean, just as Leslie talked about in her portion of the talk, we can add some different variables to studies in order to investigate some of the findings that may be not included in a traditional guideline study. We could look more at, neurodevelopmental risk and include some more of those specific type of studies. And, talking with FDA, because we're able to see a multitude of information from a number of different indications, and we can consult with different experts at the FDA in order to inform what additional information might be helpful for you to collect. Leslie, do you want to include anything?
No, I would agree with that. And just to say that, we look at literature, we look at mechanism of action of the drug, and we just go into as much detail as we possibly can to understand whether or not there might be risk to the fetus, and use that to make a decision about the clinical trial going forward.
Okay, thank you for those responses. Let's move to question number 2. So this seems to be a converse to the first question. So if you do not see a safety signal in your non-clinical studies, I'm assuming these are in the reproductive toxicity studies, is it generally safe to assume that it's all right to proceed in clinical trials in pregnant people? Or what additional information would the FDA rely on to inform on risk?
I'll jump in on that one. So what I hope we got across is that reproductive studies are designed to reveal, big toxicities. They're designed to include a lot of animals so that we can compare anything that shows up to the historical background for birth defects in that species population. But they're not highly detailed. And so there are many instances where we might not be able to really say, okay, we saw something, but we don't know exactly why that happened. And so that's a case where just because it's negative, and a lot of times when people read labels, they might say, well, there's not much in that label that sounds like it's too bad. But if you want to extend this to testing in a pregnant population, suddenly alarm bells may go off, and the reviewers at the FDA might say, well, those reproductive studies only go so far. And if we really want to define fetal risk, we have to know more. And that's where the response may come back to a company or to an investigator that says, well, we really aren't comfortable going forward until we know more about this endpoint or that endpoint, and we're going to have to ask you to do more studies.
Do you have anything to add, Kim?
No.
Okay. Thank you, Leslie.
It's never a popular response, but, but sometimes that's what happens.
No.
All right, let's move on to
investigation of that dose range is really important. and taking it slowly and making sure that you're giving the correct dose and not, it is important to make sure that the risk is low.
And I guess I'd add one more thing, which is that, companies know their drugs. And if they have done, additional in vitro or in vivo testing that gives you some insight into what risk to the fetus might be, by all means, that can be submitted, that can be discussed, and we'll take all that information in.
Okay, thank you for that. Let's move on to question number 3. And this general question was also asked by a number of audience or attendees as well. And the question is that it seems that a pretty comprehensive non-clinical reproductive toxicity testing battery needs to be completed and reviewed prior to conducting any clinical trials with investigative therapeutics in pregnant individuals. And this may be beyond the abilities of small companies to accomplish, or may lead to a delay in the initiation of clinical trials. Are there any exceptions to the drug development pathway, or is there a way to conduct non-clinical reproductive toxicity studies in parallel with the clinical trials?
This is a question we do get. I'm not surprised that it's in the chat room and here, and we get this a lot because we have a lot of really interesting studies coming in as research INDs to investigate drugs in pregnant people. And this is a hard one to answer because we realize that there is it is beyond the ability of some small research companies to have the funding to do these types of investigations. But on the flip side, if we don't have that information, if there is limited or no human data available, we don't know how to advise the dosing and whether or not it's going to be safe, and whether or not, there's going to be some risk to the mom or the fetus from a drug that may seem to be relatively innocuous. So if we don't have that data, then it's hard for us to estimate any risk. I think you have to talk with your division. I think that's the main thing we really want to get across, is that we want your drug to be developed as much as you want your drug to be developed, because we realize that this type of population is in need of information and data to inform risk. And by you talking to us and saying, this is all I've got, and this is all I can do at this point, I can't say that the division is going to be like, yeah, you can go on ahead. I highly doubt that, but we can work with you in order to determine what is absolutely needed at that juncture to make sure that your study can go forward in some respect, in order to get some reliable data to move forward. And it may just be that there's small clinical trials that go forward in a small way, and it takes a little bit longer. But that would be the advice I give right now. Talk to your division and explain, and come in with any data that you can. And if there's in vitro data, if there's literature data, it doesn't have to be this, humongous CRO company investigation, but whatever you have at this point, we will look at and review and determine whether or not it's adequate.
I would jump in and add one more thing, which is that when you're at that stage of maybe just making the move into humans, exposure data is really critical. Mm-hmm. Blood levels, tissue levels, anything that we can use to estimate what fetal exposure might be, how to compare that to the exposure in the mom. And those are studies that can be done, in vitro, ex vivo, may not require a whole huge number of animals, but that's one of the critical parameters. The first question we look at is the fetus going to be exposed? So again, you can break it down into steps so that you can make progress without feeling like you have to take on a whole entire reproductive toxicity development program. And that is all part of the back and forth in the discussion with the FDA.
Okay, thank you for that. I'd like to move into some of the questions that we received from the attendees. The first one is, how translatable and reliable are the non-clinical models for predicting the clinical fetal toxicity in different trimesters?
Do you want me to take that one, Leslie?
Yeah, go ahead.
Okay. Okay, this is sometimes difficult to describe, and I mean, inevitably the rat development process is different from a human. You know, every species has their own developmental process, and the species that are chosen for the EFD studies are typically a rodent as a rat and the rabbit, and they have been used for a number of years. So we have historical background data to inform on you know, normal pregnancy outcomes, et cetera. They are reliable in kind of giving us some information generally about what happens, you know, during organogenesis, which in the animal is, you know, what, 6 to 17 out of a 21-day cycle. So in the human, that's going to happen much earlier during gestation, you know, first trimester into second trimester. And so we're able to use that EFD study data to kind of inform timing in the human population. And it's never going to be exact. And it's very hard to say, I want to start dosing at week 18 of pregnancy. What is that going to be in an animal? And it might be like one day, you know, different from, you know, a 24-week exposure in the human. So it's kind of hard to address the timing, but we do the best that we can in using these studies to get an overall idea of, we have exposure during critical periods of development, and how can we use that data to translate into the human population. I don't think we will ever get a really definitive idea of you know, exactly what trimester is going to be affected from the smaller animal models. And it's not appropriate to use monkeys for the reproductive studies at this point, because, you know, we just don't have enough animals. It's expensive, first of all, and you don't get enough animals per litter to make any really good judgments. So this is the best we have, and it does translate. And that's why we use 2 species as well. If we don't see it in the rat, we see something in the rabbit, we're really able to kind of tease that apart and say, how real is it? Or if we see it in both, then we know it's a real finding.
I'll add one other comment, which is that one of the requirements of a reproductive toxicity program is to demonstrate that the species you're doing the testing is in fact sensitive to the drug that you're developing. And so, you know, the most literature exists on rats and rabbits and mice, but if you have to use a different species, then you use a different species. And that is something that is occasionally entertained.
Okay, thank you for that. A couple questions have come in regarding what is used to determine the safety multiples. Is it the human equivalent dose, or can you use Cmax or AUC or some other metric to compare multiples?
Well, the first choice is always the AUC. For those who know what that is, that's the total exposure for a given dose of a drug. But you may not have that value in humans, obviously, unless you've already dosed humans. So there are other metrics that are used. It can be relative to body surface area. We incorporate all that into the assessment.
Okay, next question is one that maybe some of our clinicians can give their opinion as well, but it is in regards to, is there any data or evidence out there that talks about how well the preclinical work addresses the concerns of pregnant individuals and physicians advising them? I'm assuming this is all based on the labeling statements. So we come up with labeling statements after reviewing the data, but what is the evidence that these new labeling statements are of any benefit to the population that is affected by the statements?
I think until we have a clinician that's that wants to jump in, I'll give the answer that the animal data is typically all that we have. And even in general, when we're treating not a pregnant population, the animal data is the only thing that we have to inform whether or not there's going to be a problem. And so that's when we don't have human data or very little human data, we really have to rely on what we see in those animal studies. And we try to put the best information in the label and word it in the best possible way so that the physician can understand and see what the level of risk is. And I think with the new PLLR labeling, it really helps to communicate the risk better in providing a summary of what that data means, not just here's a data dump. Now what do I do with it? The risk summary really kind of gives a better idea of, well, here's what the risk is, but it only happened at 20 times your dose. Okay, that's relatively a low risk. I think you might be okay giving it to the population. And there's also the clinical considerations section that Leslie went through that the clinicians write in saying, you know, it's better for your patient to take this drug during pregnancy than to not, because the risks of not taking it far outweigh whatever drug effect that drug is going to cause the fetus. So we're trying our very best to word it so that it is informative. And I realize that there's a lot of it's hard for us as scientists and toxicologists to directly understand what a clinician is thinking, and then vice versa. So we try our very best to integrate all the information.
Do we have any comments from our clinical people on any of the panels?
And I think it's very important to realize that the pregnancy section of labeling is written by a number of different people, not just the toxicologists. I mean, it's written by clinicians, it's informed by the clinical pharmacologists, and in certain situations, it's informed by our consultation with pediatric and maternal health. So clinicians are writing the clinician part of the label, and it's a fully integrated process through our whole team.
Yeah, hi, it's Christine Nguyen from the Division of Urology, Obstetrics, and Gynecology, and I'm an OBGYN. I would add to Kim's point that Section 8.1 is really a team effort from all the different disciplines. We do have a lot of input from the subject matter experts to phrase it in a way that could be as clinically useful as possible. The fact remains is if we have fuzzy data, there's only so clear of a message you can make when there's fuzzy data. And we do involve other divisions, particularly our Division of Pediatric and Maternal Health, to ensure that policies are adhered to and the messaging are consistent across different types of findings. So I hope that's helpful.
Mm-hmm.
Thank you for that, Christine. All right, the next question is, can modeling help out when there are gaps and extensive studies are not feasible? So I'm assuming this is in regards to what do you do when the non-clinical models aren't appropriate, or the standard non-clinical animal models are not appropriate? What can be done to evaluate the potential reproductive risks?
Well, are they talking about exposure models? I mean, certainly exposure models can be helpful.
Yeah, I'm thinking exposure models or other study models.
Yeah, well,
Maybe
yeah, certainly. I mean,
I was going to say disease models, if you could talk about those.
Disease models, sure. Again, there are some really good advancements that are going on. The amount of data that you can get from in vitro systems, the amount of data that you can get out of, genetically modified organisms. I think just the best science that you can do to inform your drug target, what your drug target in humans is going to be, how it corresponds to the animals, just as closely as you can get aligned as possible. That's information that is definitely usable.
Mm-hmm. And I think I'll point out too that there's a difference between the INDs that we receive for companies that want to develop a drug and eventually market it, and then INDs that we receive from clinical researchers who are mainly are conducting a research program and want to investigate a known drug that's being used in a different indication for another purpose. And the point of those is not always to get to marketing. You're conducting a research program. And so any information is going to be helpful. However, I think if we get to, you're going to market this drug, we need the information to inform the labeling and to inform the safety of the product. And that's why we need all of those reproductive studies up front, because we need all of the information that we can to inform the use. And it's particularly hard in a pregnant population because you're not just looking at the risk to the mom, but you're looking at a risk to a fetus. And some pregnant women don't want to be involved in an investigative process where they don't know what could happen. And some do. So it's a fine line. But any information.
Okay, I think we got time. Okay, got time for one more question, I think, and that is what can be done to encourage companies with approved drugs on the market to go back and evaluate those drugs in non-clinical or clinical studies to provide data for the safe use during pregnancy?
Oh my goodness.
Hi, this is Mukesh Saman. I'd like to jump in here. There is US pediatric legislation, the BPCA, and also the PREA acts, which are there for to answer that type of question. I'll keep it simple.
Thanks, Mukesh. So I think with that, it's now 2:15, and we're scheduled to take a 15-minute break. So I suggest that we go ahead and do that now. I just want to remind all panelists that we are still live, so please mute your audio and turn off your video until we reconvene at around 2:30-ish.
Welcome back. It is 2:30 and time to get started for our next session. We have about an hour and 15 minutes for this next session. Our next and final session for today is entitled Scientific and Ethical Considerations when Designing Clinical Trials that Enroll Pregnant People. We have a very distinguished panel to discuss a number of issues in this arena, including to discuss the ethical and scientific challenges when considering use of investigational drugs in understudied populations, pregnant people, fetuses, neonates, and long-term child development. We're going to have the panel discuss factors that impact decisions to evaluate specific therapies in pregnancy, including ethics, unmet need, benefit risk, and risk of untreated conditions. And then we're going to have the panel discuss potential designs for trials in pregnant people, dose finding, pragmatic trials, opportunistic natural history studies, efficacy and safety studies. And there have been some questions in the Q&A box about the use of real world data. We'll have the panel talk about that as well. And then discussing the timing of enrollment in terms of approaches to ensure that studies address the timing of physiologic changes in pregnancy and when to stop a trial. We're also going to have the panel discuss how patient risk preference information informs clinical trial design and regulatory review. So with that introduction, I'm going to tell you a little about each of our 6 panelists, and then we're going to have them give some short remarks, and then we're going to participate in an open discussion to further address the objectives that I previously mentioned. So to introduce the panelists, the first speaker is going to be Christine Nguyen, who actually you just heard answer one of the previous questions. She is the director of the Division of Urology, Obstetrics, and Gynecology in CEDAR at the FDA. Kathy Spong is the chief of the Division of Maternal Fetal Medicine in the Department of Obstetrics and Gynecology at the University of Texas Southwestern Medical Center. Maggie Little, is the Senior Research Scholar at the Kennedy Institute of Ethics and Professor of Philosophy at Georgetown University. Christina Bucci-Requeg is the Global Head of Pediatric and Maternal Health Policy at Novartis. Cynthia Gyamfi-Bannerman is the Ellen Jacobson Levine and Eugene Jacobson Professor of OBGYN and the Director of the Maternal Fetal Medicine Fellowship Program and co-director of the Columbia University Medical Center Preterm Birth Prevention Center. And Catherine Schubert, or Katie, is the president and CEO of the Society for Women's Health and Research. So the first speaker I'm going to turn it over to is Christine Nguyen. Christine, please.
Thank you so much, Susan. Can I have the first slide up, please? Great, thank you. Next one. Thank you. And sorry, can you go back one slide, please? Thank you. Hi, good afternoon, and thank you for joining us today. We're really excited to see so many participants, and we really hope this is part of our collaborative effort to really move this area of great unmet need along. I'm Christine Nguyen. I'm from the U.S. Food and Drug Administration. Our overarching goal is to obtain the proper data in pregnancy so that the care for pregnant people could be evidence-based. Today, I'll cover the specifics of these data needed to ensure safe and effective use of prescription drugs in pregnancy. Next, please. I have nothing to disclose. Next. So In the next few minutes, I'll cover the most important information that appears in the drug label that's been FDA approved, necessary to ensure evidence-based treatment of medical conditions, when we can apply evidence obtained in non-pregnant to pregnant people, and lastly, when missing information must be obtained from the pregnant population. Next, please. The prescription drug label is FDA's primary tool that informs essential information for the safe and effective use of a drug for a certain condition. It must be accurate and based on human data when possible. Next, please. Let's take a look at the drug label for Drug Imagine, a theoretical drug. The top 4 key categories contain the essential knowledge to properly use this drug in people, including those who are pregnant. Drug Imagine is approved for asthma and hypertension, and the dose, safety, and efficacy come from adequate and well-controlled trials for each specific use. Safety does not have to be indication specific if the 2 study populations and their dose and dose regimens overlap, and could come from pre and post-marketing sources. The last section shown there, the pregnancy section, is uniquely for a pregnant people. The human safety data in this section would be from Drug Imagines used in pregnancy for hypertension or asthma. And the non-clinical information, as you've heard earlier discussed by Leslie and Kim, would also be in this pregnancy section. So based on that, do we think we have all the information that's needed to safely and effectively treat pregnant people? A lot of it will depend on the type, next please. A lot of the answer will depend on the type of drug use scenario, which I'll cover next. Next, please. On-label use is when an approved drug is used for an approved indication. Some examples of this use include prescribing drugs to treat asthma, rheumatoid arthritis, and depression in pregnancy. Next, please. This slide shows the on-label use of Drug Imagine to treat asthma in pregnant people. The efficacy for asthma and the general safety from the non-pregnant population could be applied to the pregnant individual. The information shown in red font indicates missing or potentially missing gaps of knowledge. Whether the drug's dose or dose regimen could also be applied to pregnant people depends on factors such as whether changes in pregnancy significantly impact the drug's systemic exposure. If it does, a well-designed, well-conducted pharmacokinetic study in pregnant patients could provide the appropriate dosing facts. Although Drug Imagine pregnancy section contains some safety information, it is unlikely to be robust. So robust safety data on obstetrical outcomes such as preterm labor and maternal outcomes and fetal and long-term safety in the child are still missing when prescribing this drug on label to a pregnant person. Next slide, please. Next, I'll move on to off-label use. One second, I'm so sorry. Off-label use is when an approved drug is prescribed for unapproved use. In obstetrics, this use is most often seen for pregnancy-related conditions, such as aspirin to reduce the risk of preeclampsia and nifedipine to treat preterm labor. Next, please. So unlike on-label use, the off-label use of Drug Imagine for preterm labor comes with a lot more missing essential details shown in red font. The key evidence to inform safe and effective use for preterm labor, the dose, safety to the pregnant person, and safety to the fetus, neonate, or child, and efficacy are all not available for regulatory decision-making purposes. Because the drug has been approved, however, there is potential to generalize some safety information from the non-pregnant population to the pregnant individual, provided that the dose and dose regimen used for preterm labor does not exceed those approved. Otherwise, all these essential data must come from the pregnant population and hopefully from adequate and well-controlled trials. Note that off-label use refers specifically to a prescriber's practice of medicine in caring for an individual patient, whereas a clinical study evaluating the safety and efficacy of an approved drug for an unapproved use is considered investigational. Next, please. For the purpose of this talk, investigational describes an unapproved drug. So unapproved, and prescribers do not have access to such a drug. Next, please. Here you can see all the critical information for evidence-based use of this drug in any person, including pregnant people, is not yet available. This must come from adequate and well-controlled trials. If the investigational product is to treat a pregnancy-specific condition, then pregnant people will certainly be enrolled. But if it's intended for a general medical condition, such as severe influenza, pregnant people are not typically enrolled in the clinical trials, at least not until after the drug has been approved. Such exclusion of pregnant people may not be justifiable, especially when the disease is serious, occurs in pregnant people, and where the disease outcome could be worse in pregnant compared to non-pregnant people. Next slide, please. So in summary, certain specific information is essential to safely and effectively use prescription medications in pregnancy. And this is key to evidence-based care. One can apply certain knowledge, such as general safety and efficacy, from non-pregnant to pregnant people, depending on the type of drug use scenario. Otherwise, other essential pieces of information, including dose, must be obtained from pregnant people. Next slide, please. Next slide, please. Thank you very much.
Thank you very much, Christine. That was an excellent introduction. Our next speaker is going to be Kathy Spong. Kathy, you're up.
Thank you so much. I do not have slides. So I am delighted that Dr. Little is going to be presenting on the ethical considerations. I was asked to focus on the scientific considerations for enrolling pregnant people in clinical trials and some of the major challenges. Regarding scientific considerations, there are two main points. First, the normal physiologic adaptations of pregnancy that may impact therapeutics, and the changes in both fetal and placental development that afford different susceptibilities across gestation. Both the fetus and the placenta, of course, grow and develop across pregnancy with differing metabolism in those different stages. So first, let's talk about the normal physiologic adaptations of pregnancy that may impact therapeutics. As examples, you've got maternal physiologic adaptations, such as the doubling of blood volume, the changes in circulating binding proteins, the altered metabolism, the slow gastrointestinal transit time, and all of these may impact therapeutic dosing, pharmacokinetics, and pharmacodynamics. Similarly, the excretion of therapeutics is impacted by the physiologic changes in pregnancy due to those adaptations with the rise in glomerular filtration rate, the rise in metabolism, and again, that decrease in GI transit. So these all may provide challenges in determining the optimal therapeutic dosing in both pregnancy and lactation. Second is the consideration of the changes in both fetal and placental development that afford different susceptibilities across gestation. The changes in fetal and placental development can impact that exposure, and that exposure is dependent upon the timing of that exposure. Generally, at the earliest stages of pregnancy, the impact of something that could be teratogenic could result in pregnancy loss. If exposed during organogenesis, generally considered the first trimester, you might result in a major structural abnormality such as spina bifida, a heart defect, or cleft lip or cleft palate. Whereas exposure later in pregnancy tends to impact organ differentiation, growth, or function, such as hearing loss or microcephaly or cognitive impairment. So this is just one concrete example. Maternal alcohol consumption in the early embryonic period is associated with structural birth defects, impacting the facial appearance, the brain, and growth, whereas consumption later in pregnancy is more associated with behavioral disorders and impaired cognition, but without specific structural abnormalities. I wanted to spend a couple of moments on the major challenges for enrollment, as I was asked to do. And those I kind of align into four main areas. The first is the importance of buy-in from both scientific and pharmaceutical communities that inclusion of this population is critical, as without their inclusion, we have no data on the effectiveness or of the risks of that therapy. This essentially results in experimenting every time a therapy is administered. At present, there's essentially a default to exclude both pregnant and lactating women from research trials. Even with decades of recognition of the importance of their inclusion, I think the COVID therapeutics and vaccine development again excluded these populations. Another major challenge is that concern of liability. Without strategies to mitigate or absolve litigation, this is going to remain a major challenge, and it's likely that pregnant women and lactating people will continue to be excluded. This results in a very difficult situation. And I do think it's important to remember that therapies that achieve FDA approval in reproductive age women are approved for that same indication in pregnant and lactating women. So they're not being used off-label. But without data, our professional societies must rely on expert opinion for their guidance for their use. A third challenge is the education of the communities of the importance of this research. There is a robust history of doing research in these populations and in creating an evidence base for practice. But just like any research, it requires buy-in and education of the community. And I know that Dr. Jomfy is going to be talking more about that robust history we have. And then lastly, the financial support to conduct this research. Funding for these studies is mainly limited to government and NIH support, and that is extremely limited. I did a quick NIH RCDC coding look at the funding of pregnancy research, which has ranged between 300 to 500 million for the last several years and represents 0.2% of the NIH RCDC funding. So I thank you again for this opportunity to discuss these topics that I am passionate about, research and the inclusion of pregnant and lactating women in research. We do have decades of experience and success in their research, and it's important and critical to move beyond studies focused only in those groups and to include them in critical studies that will impact their health. Moving to assuming inclusion of pregnant and lactating people rather than exclusion and protection through research is critical for their care. Thank you.
Thank you so much, Kathy. Really appreciate your comments. And now we're going to hear from Maggie Little. Maggie?
Thanks, and I also don't have slides, so if you want to switch back. And I'll just say at the start, anytime my presentation bears any resemblance to something Kathy Spong, who's a goddess, does, it makes my day. So I've been asked to speak to the ethical considerations and challenges in enrolling pregnant women in research. And I come to this after 15 or 20 years of advocacy on this issue, and as a member of both the Prevent and the Phases projects, which gathered working groups and established guidances on how to do exactly what we're talking about today in both emerging pandemics and, again, the HIV, TB, malaria space. And I'll chat the link to those guidances. I want to zoom way out. When we're talking about ethical considerations for enrolling pregnant women, there are really two questions. Two types of issues. One is, when is it ethically permissible to allow or pursue enrollment of pregnant women in a given trial? When are we, when ethically and under the regulations, may we include them? The second question is equally as important. When do we have an ethical responsibility to allow or pursue pregnant women's inclusion in a trial? So if it were permissible to do it, do we have a responsibility to do it? And it's very important to keep both of those questions in mind. Let me just start with the first one. So when is it ethically permissible to allow enrollment? We've heard lots of details, and really the devil is in the details on this stuff. And I can listen to my FDA colleagues all day long, so thoughtful about the challenging questions about interpreting preliminary data into a sort of entrance ticket to doing the next level of investigation, entrance ticket into the next, right? But pulling back, there's broad ethical consensus as well as regulatory consensus that there's sort of two pieces to this permissibility question, okay? The first one is, do you have preliminary evidence that gives you some signals about potential safety concerns. Lots of complexity over how robust the evidence needs to be. We've heard lots of fabulous details about that. And how should we set the evidence level before we allow inclusion? After all, there can be a catch-22 with research with any human, right? If you ask for too much preliminary before you allow enrollment, you never get to do the enrollment, and then you never get to have well-designed, highly regulated and responsible research to answer questions. Same happens with pregnant women, okay? But at any rate, you can't go in blind. It's not enough to say there's no evidence of danger. If there's no evidence that allowed the chance for dangerous signals to arise, you can't enroll women, pregnant women, okay? And that's consensus on ethics. Everybody agrees. We want to have specific protections here. A challenge has been mentioned already, but getting that preliminary evidence early enough in drug development, since it is a precondition. If you don't have that, you don't have an entrance ticket, right? So the more we can move that up, the more we can have potential, the possibility to enroll pregnant women in even drug development trials. The second piece of eligibility is, can you meet the standards of research-related risk that would be represented in participation? So based on the preliminary evidence you have, right, that's the first part, which won't give you any guarantees, won't even give you statistical evidence, right, but it's suggestive evidence. Do you think the potential research-related risk to woman or fetus, or both, is acceptable? And here the regulations and ethics are kind of in agreement. There are two kinds of standards. It's a disjunct. If the research carries no prospect of direct benefit for either the woman or the fetus, okay, then the research-related risk to the fetus has to be capped at minimal risk, which is, by the way, a crappy risk standard nobody likes, but it's what we've got. It's hard to figure out a better one, okay? This is very unusual when there's no prospect of direct benefit. So less important to figure all of that out, but important to know. The other standard, though, is for trials that offer any prospect of direct benefit to either the woman or the fetus. The allowable research-related risk standard, even to the fetus, is a ratio. We can tolerate more potential risk to the fetus if there's more potential benefit to mom or fetus, okay? Very important, because that point gets really confused often. Many trials where there's people will tend to only look at the possible risks of participation, but you have to look at risks in relation to potential benefits. Otherwise, you actually can do a disservice to disallow pregnant women. It might have been beneficial on balance. They're getting included. Let me switch to that second question about when does a trial have a responsibility to include pregnant women? So obviously, it would only be if it is permissible, but right now there's this incredible gap. Trials where it would be permissible to enroll pregnant women, meet all those conditions I just talked about. Nobody's making you do it. Nobody's saying you even have to presumptively do it. This second question is about, if it were permissible, how strong is the should to pursue it? How do we discharge our collective obligation to get an adequate evidence base for this subpopulation? And the key challenge here is not an ethical one, it's a structural one. Currently, that collective obligation to gather an adequate evidence base is undistributed. It's nobody's job right now. It's not even anybody's presumptive job right now. So one of the key things that Preglac and others are looking at, the guidances I talked about, are talking about how to do some structural changes that are still thoughtful. It's not the case that every trial needs to enroll pregnant women. Right? It depends on lots of factors. How significant is the disease and prevalence in pregnant people? How robust is the evidence that you could add to the pot if you allowed enrollment of pregnant women? Are there relatively easy available inclusive designs that don't add tons of money, but would give us usable data, especially if pooled with other people's data, right? Some of the key issues are, as our fantastic FDA colleagues have talked about many times, could the FDA distribute some of that obligations in the pre-approval space to say pharma needs to pursue more. And they're, in my humble opinion, they're doing an amazing job of really pushing this issue and advocating. And we should feel for them because they have limited legislative authority right now to require a lot. So the structures that need to be changed may be enabling FDA to do things that we would love for them to do. A second one is large funders like the NIH. So my understanding, and NIH colleagues, please correct me if I'm wrong, but my understanding is that when NIH does funding for large clinical trials, like phase 3 clinical trials on important medications, they say that if you're going to get an NIH grant, inter or extramurally, you have to justify excluding, right, from the general population, people with comorbidities, kids, elderly, minorities. This, by the way, is for post-approval research, which is often very key. Pregnant women are missing from that list. It's the only subpopulation that's missing from that list. So we could just please add that you need to, maybe you have justification, but you need to think about it and presumptively include pregnant women, because if it's a large clinical trial of an important drug, pregnant women are amongst the end users. They might be amongst the most important end users. So you need to think in designing your study about whether they need to be included. And then just a shout out on one of the challenges, again, not ethical, but logistical, for individual trials that are really trying to do the right thing and be part of the solution, to adapt their designs to allow the eligibility of pregnant women, to reduce the barriers to pregnant people's inclusion, which include kooky contraception requirements. They're good ones, but they're kooky ones. Or again, to just automatically use pregnancy as an exclusion criteria without. But one of the biggest challenges to those good actors who are trying to be part of the solution really is about trial insurance. So unless we do better at just making it easier to find a trial insurer that will allow you to enroll pregnant women and will not hike up the rates crazy, right? But we just need a consortium of willing trial insurers who understand this issue. And that would open the door to the really significant number of researchers and trialists who want to make progress here. Thanks.
Thank you so much, Maggie. That was an excellent talk, and I think segues very nicely into the next comments by Christina Bucci-Requeg. Christina, you're next.
Great, and I also do not have any slides for today. So I will be speaking to you from my perspective from the industry. Just to understand where I'm going to be spending my time today in my prepared remarks is I was asked to speak a little bit about providing industry insights into how we think about inclusion of populations in our trials. So by way of background, I come to you with 10 years of experience in the research policy space specific to pediatrics and maternal health, and before that, spent 10 years in research and development specific to pediatric and women's health drug development, both of which in those 10-year periods were at Novartis Pharmaceuticals. I also am one of the co-drafters for the European Forum's Good Clinical Practice position statement on the importance of inclusion of pregnant women in research, and also have contributed as an industry collaborator on the HHS task force on research involving pregnant women and lactating women. So this is a passion topic for me, to which I spend my day looking to find ways to advance the research policy arena for pregnant women. So just to understand where I'm coming from at this, also from the standpoint of the pharmaceutical industry. Now, I work within the innovative pharmaceutical industry. So what does that mean? Our research investments in the innovative pharmaceutical industry are in investigational drugs and biologics. So when our products' patents expire, our research labs close. So when we're talking about the research that we work on, we're not typically talking about repurposing and opportunities to better evaluate those standards of care that have been in the clinical care space for decades, but have still as yet an inadequate set of information to inform on the appropriate use in pregnant populations. So in my space, typically we're talking about a very fast pace of technological advance. Think of the current pipelines of today, cell therapies, gene therapies, CRISPR technologies. We also typically, we're not fully funded to have expertise in the entirety of the research space. So we rely on expertise and we rely on our collaborators. These stakeholders that are in this session have been on the call today to be able to execute fully on our non-clinical and clinical programs to inform on our product development. And additionally, we rely exclusively on well-staffed, well-researched, and funded research infrastructures to be able to be carrying out good laboratory practice and good clinical practice related trials. In addition, as I mentioned previously, we focus entirely on the investigational space. Now, the key influences that underlie our general investment in innovative products or in investigational products come from two different arenas. One is on the demand pull side, meaning there is a commercial marketplace for there to be development for a specific disease or a condition, or on the technology push side of the space, where there is clearly a technological advance that allows us to better research and evaluate a disease or condition. And without these two key conditions, the demand pull or the technology push, there typically is an absolute and direct impact on our investment in innovation. And so this could, if you are lacking the demand pull or the technology push, completely erode how industry invests in certain innovative technologies. So as a result of this, we have seen that over the decades, there has been certainly a lack of investment in the development of therapies specific to pregnancy condition. In addition, there are, just as all of the speakers before me have spoken, clear evidence gaps in our product labels on how to inform on the appropriate use of therapies in pregnant populations. So what are the factors that are influencing this? And how are these factors coming from how investigational, innovative pharmaceutical companies are designing their clinical trials? And I will say, to ground all of this in much of what was said from session 2 before us, that much of what we do in the investigational space is entirely grounded in the benefit risk at the time that our trial is initiating. So if there are large data gaps, there are large sets of information that are unavailable to us to inform on the appropriateness for inclusion of certain populations, then those populations will not be able to be studied at that time. So I typically think about this from 3 different perspectives, from this perspective of how we put information into how we're designing our protocols, also how external influences such as the IRB review may influence whether or not those protocols will be acceptable, and then finally on that operational component of trial research infrastructures. And so when we go back to protocol development, you heard in session 2, very extensively covered by Dr. Hatfield. There's broad international consensus on the regulatory standards to evaluate reproductive toxicology and fetal development. And this has come through international consensus, largely through the ICH. And these non-clinical studies are a routine requirement for all of our investigational products that we're evaluating. So this is something that the pharmaceutical industry is very knowledgeable about and works very collaboratively with non-clinical research laboratories and the regulators on a daily basis as it relates to our products. In addition, these guidance documents put together through international consensus include extensive consideration in regards to the appropriate timing of these non-clinical studies for our investigational products and where they're at in their development stage. But I do think there are ways that we can broaden our thinking on how to better leverage this information. Not only this information, but other animal data for incorporation into pharmacometric models that could better inform on dosing when we're talking about physiologic changes during the course of a typical pregnancy, and also how that information could potentially better inform our trial design decisions, such as how we include specific populations, and in this case, pregnant populations. But as stakeholders, we need to get together. And when I talk about stakeholders, I'm talking about patient groups, sponsors, agencies, and academicians on how we can better leverage existing product and class information that might help us better inform on dose selection and trial design considerations. And in addition, there is a clear need for better leveraging of real-world evidence from pregnant populations within the disease of interest to be studied to help us better understand relevant endpoints. We don't have good understanding of relevant endpoints during pregnancy, not only for the patient, but also for the fetus, so that we can better inform the type of endpoints we need to assess, the kind of outcomes we need to assess for both of these populations, not only as it relates to the efficacy of the product while it is being utilized during pregnancy, but also to the safety. So how can we better collaborate to develop and enhance existing models that will more predictably inform our ability to include pregnant populations within our design? Additionally, now when we turn to the influence of external factors such as IRB review, you've also heard in session 2 in some of the discussion that in early phase investigational drug research, it is commonplace, or should I say the rule, for pregnant populations to be excluded from research. And it's solely based on the basis of what we know and don't know about the mechanism at the time that trial is starting. And given their construct and interrelationship on the benefit risk between the pregnant person and the developing fetus, there's not currently an easy answer to rectify this, as we need data to better inform the appropriateness for inclusion of the population in a trial design. So what we need to think about is how better to leverage any information that we have through other potential means. In later research, it's incumbent upon all of us stakeholders to more positively favor inclusion and or more positively speak to the appropriateness for exclusion, or to demand an understanding of a clearer understanding, for the exclusion of certain populations. And in addition, we need to align on what is an appropriate sample for inclusion in the population to adequately inform on the efficacy and safety that will inform on the appropriate information to include in a label. And then finally, as it relates to trial research infrastructures, we have thousands upon thousands of molecules that are in our pipeline. So it is important for the academic research community to be able to answer the question about infrastructures and whether or not they're adequately funded to be able to conduct the type of research that would be necessary for every molecule within a pipeline. So these factors alone or combined have important and variable impacts on our decisions related to inclusion. So what I would like to say in summary is that within the existing regulatory framework, it is truly the minority of products that are authorized with a label that includes warning and/or contraindications for use in pregnancy. However, with this being said, I fully acknowledge that too often our products that do cover pregnant populations within the approved label do have limited clinical information to inform on the best use across the trimesters of pregnancy. And in addition, I fully acknowledge that patients should not have to shoulder all of the risk in taking a product whose benefit risk profile is not well understood when the product is intended for their use. So given all of this, it's incumbent on us as the stakeholders represented here to work together to find pragmatic solutions to enhance our understanding of therapies that are intended and prescribed for use in pregnant populations. So thank you for your time.
Thank you so much, Christina. We're going to move now to hear from Cynthia Janfi-Bannerman. Cynthia?
Hi, I'm Cynthia Janfi-Bannerman, and first of all, thank you so much for giving me the opportunity to speak to you all today. Next slide, please. So I was asked to give specific remarks on how we apply ethical and scientific principles to different clinical trial designs to meet the trial's objectives. And I'm speaking to you today as a clinical trialist. So one of my hats is being the principal investigator for the Columbia Center for the Maternal Fetal Medicine Units Network. And throughout the course of the talk, I'll explain to you what that is. But I think the very first thing to say, I know this is a mixed audience, and like Dr. Sheffield mentioned before, I am preaching to the choir, but in fact, we do very regularly and routinely do clinical trials in pregnancy. And so I'll go through what some of those are. Next slide, please. So first, my disclosures. I do have funding from NICHD and NHLBI. I have a grant from Hologic to study preterm birth, and I'm a member of the New York SURA Advisory Board. Next slide, please. So there are two broad categories when we're thinking about clinical trials and pregnant people. And the first one can be maybe even subdivided further into two topics. So first, there are interventions to improve pregnancy outcomes that are very specific to pregnancy. So preterm birth, preeclampsia, cholestasis of pregnancy, these are conditions that happen within pregnancy. And so there's really no need for pharmaceutical companies or really anyone to study them outside of pregnancy. And within those are drugs that are used kind of specifically for pregnancy, and then drugs such as perhaps a statin that is commonly used outside of pregnancy that might have a pregnancy indication. And I'll explain that further in a second. And then the other broad category would be interventions of common medical conditions that actually coexist with pregnancy. So for instance, hypertension and pre-existing diabetes are present in 5 to 10% of pregnant women. And so they become pregnant, they need these medications, and what is the guidance that we have? And I think several folks have touched on how we approach that in pregnancy. I think involving them initially in clinical trials is one of the ways to consider moving forward. And then I think very remarkably, I think everyone has seen what has happened with the development of a COVID-19 vaccine that excluded pregnant women in clinical trials, specifically because pregnant women become more sick, is what the early data show. Pregnant women become more sick when they get COVID-19 than their non-pregnant counterparts. So pregnant individuals were left out of these trials, and over 70,000 people were enrolled. So it does leave, as a clinician myself, a clinical conundrum in terms of what do we do with the new information since pregnancy was an excluded category. Next slide, please. So one of the answers is the NICHD MFMU, and I can tell you a little bit about the origin. For many reasons that are historic and kind of beyond the scope of what I'm going to tell you today, obstetric management for high-risk women specifically had been adopted based on best practice, and whatever your senior person did, you also did without the best evidence, again, because there was hesitation to do any type of clinical research in pregnancy, and that's historic and paternalistic at best. Next slide, or next, yes. So to address the need for well-designed clinical trials and in maternal and fetal medicine specifically, the NICHD established the Maternal Fetal Medicine Units Network in 1986. Next slide, please. So these are the centers. There are 12 centers currently in the current cycle, which is about to end in a few months around the country. And like I mentioned, I'm at Columbia University and the principal investigator from that center. But you can see there's a geographic and racial and ethnic diversity in the centers that are represented here. Next slide, please. So it's the only federally funded obstetric clinical trials research network. And what we do as clinical trialists is we conduct clinical studies to improve maternal, fetal, and neonatal health. The greatest priority of the studies that we do are given to randomized trials with an aim to reduce morbidity to mom and baby related to preterm birth, fetal growth abnormalities, and maternal complications. We also are addressing maternal mortality in very new and significant ways. We're providing rationale for evidence-based, cost-effective obstetric practice. Next slide, please. So I'm going to give you an example of a study that has kind of been ongoing, both in the Maternal Fetal Medicine Units Network, but also started in the Obstetric Fetal Pharmacology Research Unit. So there is a pharmacology unit that looks at medications in pregnancy specifically. And now several years ago, there was the initial desire to look at pravastatin for the prevention of preeclampsia in high-risk women. So this was a pilot study that was done in that center. Next slide, please. So the primary research question here was, what are the pharmacokinetic properties and maternal and fetal safety profiles of pravastatin when used as a prophylactic daily treatment in pregnant women at high risk of preeclampsia? And there are animal data and hypotheses that suggested that pravastatin would actually be excellent at reducing recurrent preeclampsia in women who are at high risk. Next slide, please. And so the outcomes of the initial study, and you can see there were only 20 women enrolled because this is a pharmacology study, and so they were looking at safety as one of the primary issues, but also efficacy signals as well. And you can see, if you look at the top bar here, that all of the recurrent preeclampsia occurred in the placebo group. In fact, there were no cases of preeclampsia in the pravastatin group. So highly potentially efficacious for one of the top 3 causes of maternal mortality in this country. You can also see that blood pressures were a little bit lower, length of hospital stay was a little bit lower, but very, very promising. However, we are still in the process of trying to get this studied kind of in a larger scale group because this drug needs to go through the processes of the FDA. Next slide, please. Next slide. So there is a planned randomized controlled trial of pravastatin for the prevention of preeclampsia in high-risk women on a larger scale so that we can confirm the efficacy seen in the prior studies. But again, like I mentioned, there was a second pilot that was done, again showing efficacy, again showing no potential risks to mom or developing fetus. And ultimately, we do know that the risk of preeclampsia are quite high and extend into programming of the fetus and into childhood. And so hopefully we can get drug or trials like this completed, but you can see how with the FDA process and with everything that was described earlier and how drugs go through the FDA, it's taken us quite some time to get this done. And potentially there's thousands, hundreds of thousands of women that can benefit. Next slide, please. So I'll also talk a little bit about how the MFMU has changed lives through another drug study. So in fact, it's called the Eunice Kennedy Shriver's NICHD, which is named after JFK's sister. Next. His first son, Patrick, died of respiratory distress in 1963, and next. And he was 35 weeks gestation. So one of the studies that I was involved with looked at betamethasone for fetal lung maturity and just maturation of the fetus in general to prevent this. Interestingly, and it was alluded to, I think, in several of the talks, betamethasone is FDA approved for other indications. It is not approved for use in pregnancy for this indication, and it is used off-label. And oftentimes, as practicing obstetricians and as clinical trialists, we can say that sometimes we're unencumbered by the need for FDA approval because we can use some of these drugs off-label, and the processes might take quite some time to get them approved. Next slide, please. So this was the results of the study that we did. Again, using a drug that is essentially off-label use, and it helped to reduce the risk of late preterm respiratory morbidity, but also it's used from 24 to 36 weeks and very beneficial. One of the most commonly prescribed medications in pregnancy in the treatment of preterm birth, and again, kind of off-label use. Next slide, please. And importantly, so the studies that we're doing in pregnancy inform our clinical practice. So you can see that the guidelines in pregnancy surrounding the use of antenatal corticosteroids are based off of these trials. So, the question of whether women want to participate in these trials, there were 2,800 women that participated in that particular one. We are doing clinical trials in pregnancy that inform our guidelines, and we think it's very important for folks who are doing clinical trials on drugs where pregnancy might not be the primary indication. I think it's important to include them because we do need evidence-based data for what we do for pregnant women. Next slide, please. So ultimately, I was asked, how do we apply ethical principles? So first, there's informed consent, and this is obviously a very important part of clinical trials. Very smart people have asked me, oh, but pregnant women don't participate in clinical trials. And this is just not true. My entire career would be over if that were the case. And if you approach a pregnant woman for a clinical trial, what we have found is that about 50% will consent. Some people don't want to hear at all about research. Some people are very interested. 50% is about average, and it also depends on what you're studying. There's a clinical trial we're doing now with one of my fellows where it's about a 90% consent rate because essentially we're just studying timing of a medication and randomizing it into that. So there are women who want to participate, and with the informed consent process, you can then find the women who are interested in learning more and helping people in the future. We publicize trials to allow participation of pregnant individuals and those who are potentially eligible. And an example of that is the CMV study that we recently completed, where we put out on social media, if you are interested or might have a CMV infection and are near one of these 12 centers, please come and enroll. And so it is important to try to get as many people who are interested the ability to participate. In a clinical trial setting is probably the safest way that we can introduce medication in pregnancy because of the monitoring for safety and for SAEs that are reported within 24 hours to the sponsor. And it's, I think, much better than the alternative of saying, why don't you go ahead and try it and put your name in a registry? Because that is just not going to give you as robust evidence. And I would argue that it would be unethical not to study promising interventions in eligible pregnant individuals. Next slide, please. So it was brief, but I'll be happy to answer any questions at the end when we go into the Q&A. And thank you again for the ability to present today.
Thank you so much, Cynthia. Great comments. And then finally, we're going to hear from Catherine Schubert. Katie?
Thanks so much. And I will be pretty quick because I know we have a really esteemed panel and there are questions and comments, and we've all been working together for a long time and having this discussion over many years. So just by way of background, I'm Katie Schubert. I'm currently president and CEO of the Society for Women's Health Research. And there we leverage science and evidence base to ensure that patients are empowered to take charge of their health care. Before SWHR, I was at the Society for Maternal Fetal Medicine, where I largely took on the issue of inclusion of pregnant people in clinical research. And so my background is really in the policy and advocacy space, as well as coalition building and sort of how do we come together to move the needle forward. So I just have a few comments I wanted to make from that perspective, and they're not going to be anything that you haven't already heard today, because it's sort of a combination and a collection of everything that those who have spoken before me have already said. And so I think importantly, I want to really thank the FDA and the group of panelists for this public meeting. It's so important, I think, to have this conversation and to really share ideas, particularly during this time when, we're in the middle of a global health pandemic. We are seeing firsthand how either a lack of inclusion or need for more information or data is really critical to ensuring that people are able to get the care that they need. And so I think there's a couple of things regarding potential strategies that we could use to sort of advance the idea of, the culture, including pregnant people in research rather than excluding it. And some of those might be more shorter term and some might be more longer term. And we've seen that sort of through both the conversations today, but also in previous conversations at the Preglac task force meetings. So I think ultimately, without strategies for inclusion, the burden really falls to the patient and their provider to kind of work through what works best for them and sort of what information they have available to make decisions. And it's really with, in many cases, little evidence or data to be able to make those decisions. So from that standpoint, I think, again, as many folks have said, we really do have an obligation to sort of shift this culture to inclusion rather than exclusion and build this population into study goals from the beginning. And I would say not just looking at the pregnant population, but thinking about how we can include different races and ethnicities and geographic locations and other areas of need to really make sure that the trial populations reflect the broader treatment populations. We have pretty significant health disparities in this country, particularly when it comes to maternal health. And so in an effort to make sure that we're trying to tackle those as best we can, I think that that's a really critical strategy for us to kind of start with. Beyond that, I would say sort of thinking about the conditions that someone might have prior to becoming pregnant. And so we've talked a lot about studies that might happen to look at conditions specific to pregnancy, where we know, there are already research opportunities available and there are models to look at. But what about those areas in which someone might require a medication to treat some sort of condition during their pregnancy that they might be afraid to continue using during that time? So I think that's a huge area of need and an area that could be focused on in the shorter term, as well as sort of looking at better education and awareness, not just of trial participants, but also of clinicians and researchers. So just real quickly, learning from what we've already done in this population. Cynthia just mentioned this, Kathy's mentioned this, others have talked about this. There is data that exists out there. So how do we pull from A, the models of research that we're using, but B, also that real world data that Christina mentioned? It does exist. We know that people are taking these medications during this time. So I think there may be an opportunity for us to be able to collect some of that data and really move the needle forward. In terms of sort of looking at some of the things that were mentioned, the common rule was mentioned, first thing today. And I think that's something that not a lot of people are really talking about. Pregnant people are no longer classified as vulnerable. And so I think we can spread the word and be thinking about sort of how do we better include that population as a key strategy, as well as the consent issue, which was mentioned. But I think that's another thing that came out of Preglock, really thinking about the strategy surrounding consent for inclusion and thinking about how maybe we can streamline that a little bit and make it a little bit easier for folks to enter into a clinical trial or research. We talked a little bit about the training, education, and awareness piece for both the sort of folks entering a study population, but also I would say on the workforce side. So how do we really shore up the desire for people to enter this field and do the work that, Cynthia and Kathy and Christina are all doing, because it is really an area of need to have that workforce up and running and moving forward. And then also this cooperation and collaboration piece, which you've heard a lot about. We've heard about public-private partnerships. We've seen, a model in the EU where they're sort of working with industry and government to be able to move the needle forward here. There are models that exist that we could probably look to really divvy up some of the responsibility among, members of the clinical side, researchers, industry. We all play a critical role in making sure that the conversation continues and we can move forward. I couldn't not mention de-risking the research. I'm really glad that Maggie had a chance to speak, and I know we'll be talking a little bit about this probably later and also tomorrow. But this is a really huge barrier for changing the culture of inclusion here. And it's not easy, but we need to be able to tackle it to be able to move forward on this issue. So, there's tough topics like liability reform, determining key incentives or mitigation factors for industry and other researchers, and maybe even thinking through some sort of alternatives for that, I think is really going to be important for us to move forward, and that time is probably now. I think we've had a lot of conversations leading up to this, and we really need to tackle that piece. I know, Erin, you mentioned the Preglock implementation report. On the advocacy side, we're looking at that really closely to make sure that we can support these conversations moving forward, because I think that that's probably the next best step for us to be able to really push for broader, a broader culture of inclusion. I mentioned a little bit about the real world evidence, but also these ideas of data sharing and access and infrastructure. So, we started out the day seeing the FDA Pregnancy Exposure Registry website. These data sources exist. And so being able to leverage what already is there and the infrastructure and study infrastructure that we already have is really going to be critical. And I think a key strategy, at least in the shorter term, to be able to start moving the needle forward on this. And really thinking about how we can use this roadmap to really start from a place of inclusion of, this population across the board, so that we can really tackle some of the pressing issues. And I think the biggest thing, from my standpoint, and Cynthia mentioned this, is that COVID has actually presented us an opportunity to have this conversation. And so it's an exciting time. So thank you very much.
Thank you very much, Katie. And I think we're going to get back to some of the concepts that you mentioned. So what we're going to do, first off, I want to thank all of the panelists for their really insightful comments. We had come up with a couple of discussion questions. We're going to move into the moderated discussion portion. I apologize. The wonders of working at home. My dog has been asleep all day, I swear. So, pardon me for just one brief second. Pardon me. So now I'm back. So Yes, it's my little Dusty. He loves to do Dusty Snow Angels. We are doing the moderated session. I'm sorry that we don't have more than about 15 minutes because I want to be mindful of everyone's time. We have a few discussion questions that we came up with, but I want to remind everyone in the audience to feel free to submit any questions to the panelists via the Q&A function located at the bottom, and we've received a lot of really good questions, so I'll pull from those in just a minute. And just so you know, the Q&A function is at the bottom of your Zoom screen. And then for all of our speakers who want to add a comment or question, please add that into the Zoom chat box, and please send that to all panelists and attendees. So I will try to keep an eye on that as well. Just to read through the discussion questions, and then I'm going to have everybody come up on the panel so I can actually see people raise their hands either virtually or actually. The discussion questions that we had, I'll read them, and then I want to focus on number 3 and number 5 just to give everyone a heads up. So the first question is, what information regarding drug therapy is typically needed and can only be obtained in pregnant people? And then what are the ethical considerations and scientific considerations for enrolling pregnant people in clinical trials, and what are some of the major challenges? And then the one I want to focus on first is discuss the principles such as the severity of the condition, unmet need, benefits, risks, and uncertainties that support enrolling pregnant people in clinical trials. And this should include the risks of an untreated condition and the value of the information that could be obtained with research. And then discuss the timing of enrollment and when to stop a trial in pregnancy. And then number 4, how do we apply ethical and scientific principles to the different trial designs to meet the trial's objectives? And there was an ethical question in the chat box that I know, Maggie, you responded to, but I'd like to actually have you respond out loud to that because I thought that was a really interesting question. And then what strategies could be used to advance the culture of including pregnant people in research? So what we're going to do now is we're going to take those questions down. We're going to bring everybody up. There we go. And I can see everybody. So that's fabulous. If you want to, speak or raise your hand, I will try to see you either visually or virtually. The first question is that I'm going to have people talk about is the principles such as the severity of the condition, unmet need, benefit, risks, and uncertainties that support enrolling pregnant people in trials, including the risks of an untreated condition, which is, I think, something that we haven't really focused on a lot today, and then the value of the information to be obtained with research. So who wants to go first? Okay, Maggie, you're up.
I might start us off. So I think that list already names lots of the key issues, right? What's the severity of the disease we're trying to prevent or treat? What's the incidence, the prevalence of it? Is it worse for pregnant people or not? But pulling back more broadly, when we're trying to prioritize ensuring that we have inhuman data in pregnant people, we're looking at 2 different risks we're trying to mitigate. One is the danger of there being unknown safety risks, right, to either the woman or the child that might go unnoticed when we do roll out the medicine and pregnant women use it. Because as many of the speakers said, pregnant women do have underlying or emergent health issues. They need to use medicine. They do use medicine. Okay. So one is, when do we need to, what are medicines pregnant women use a lot that really might carry unknown risks to the mom or baby, either through toxicities of the drug or through under or overdosing, because we don't have the dosing information. But the second risk we're trying to mitigate is actually the opposite of that one. Where is a lack of data erecting barriers to pregnant women getting needed medications? And unlike some subpopulations, it's that second one in our view on the phases to prevent that there's actually the bigger risk that pregnant women face, right? So teratogenicity is actually rare, thankfully, right? It's really that there's several disease states where pregnant women are relegated to sort of last in line to receiving game-changing new therapeutics, vaccines like with COVID, or preventives like PrEP and HIV, because of a lack of evidence. So there's reticence, either the woman herself, understandably worried because she asked her doc, and the doc says, we don't know, or the provider never even offers it because the provider has a reticence, or especially in low-income settings, guidance setting entities like the World Health Organization are, even if the drug is approved for use in pregnant women, are often reticent to put their stamp of approval in saying we recommend this for use in pregnant women until there's any human data. So part of what we argue is with the prioritization is really looking at where data is needed to remove barriers of access to life-saving medications.
It looks, Kathy, you look like you had wanted to say something.
No, Maggie's covered this beautifully. I think it's very difficult, given the dearth of information we have on most medications in pregnancy, to be able to say which ones are the ones that we need to study, right? And I think it's part of it is burden of disease, right? So where are we, if we studied something, where are we going to make the biggest impact? But part of it is these new therapies that we don't have data in pregnant women, so we use something that is perhaps suboptimal. And I think one of the key things that people don't necessarily recognize is the reticence of physicians to change doses, or of patients to change doses, recognizing that they may need a higher dose in pregnancy, but they are afraid to take that higher dose because they want to minimize their exposure, but they're already exposed to that medication. And if it's not therapeutic, it's not going to be beneficial to them.
Thanks. Excellent point. Anyone else want to weigh in on this particular question?
Yeah, I mean, I might add that there's two issues at hand. One is the prevalence of the disease and how common it is within pregnancy, but then the other is the severity. And the things that kill pregnant women are the things that we should probably spend most time studying. And with preeclampsia, we have this emerging treatment that's potentially helpful, but is waiting for that kind of perfect teratogenicity study that says that, you know, it's going to be okay. I think Dr. Little said it the best. Teratogenicity is not that common, but death from preeclampsia is. And so kind of risk-benefit ratios are going to be really important in how we address research in pregnancy.
Yes, Christina.
I want to take it in a slightly different direction. And I think Maggie actually hit the nail on the head. It's all of these pieces that are going to help us really define the directions we need to go in. But we have to do that difficult task. We really have to have this difficult conversation because there's not enough resource to cover everything. So where is the clear need, and where are the low-lying fruit opportunities where we can really make big gains by focusing in those areas, by directing research in those areas, by directing interest and investment in those areas to be able to answer those questions, because then we begin to address pieces to be able to then move on to the next set of priorities. So we have to we talk a lot about prioritizing and how difficult it is, but we just need to sit down and say, here are going to be the things that we are going to prioritize and do that difficult task to be able to get there.
Yeah, Christine.
Yeah, just follow on Christina's point about low-hanging fruit. I mean, to me, we have so many medications approved in adults, including pregnant adults. They're being used every day. Where, you know, it's not contraindicated. We're having really a vast opportunity just to get pharmacokinetic data in these drugs. And even those are not being conducted. And for in those situations, to me, the research risk is just extra needle sticks, right? Women are pregnant, people are receiving them as part of their care. So can we even start there? And if we can't, why not? I mean, that will make me very sad if just that very simple research is not done more, particularly for the drugs that are used most commonly in pregnancy. You know, the systemic drugs are used in pregnancy. So, I mean, I would propose that as a starting point. There's very minimal risk to even obtaining those data. And those data actually are critical because we're finding the exposures are similar or we're having to adjust the dose. That's how we do evidence-based care. We can tell a pregnant person, listen, the dose you're on is what you're supposed to be on. And, you know, if you don't have benefits, there's no tolerable risk, right? You got to have benefits. So if you're getting sub-therapeutic dose, then whatever risk you're taking is really not worth it. So I'm just trying to introduce some very simple ideas that I think we can start tomorrow.
Thank you, Christine. And I know, Kathy, I know you want to get in here, and I'm going to expand the conversation to include real world data. Because you know, you were talking about, you know, PK studies but are there ways that we can include additional real world data that might be able to help inform either labeling or better use? So Kathy, you don't have to go there. You can go back to the original question.
Oh, it's all the same. It's all the same. I thank you for that. And Christine, you're absolutely right. I mean, we give medications all the time, some on label, some off label, as Cynthia mentioned. And we give them because we have evidence in some cases that those medications are helpful, right, and are beneficial, but we don't have the PK/PD data on pretty much any of them. And all of those physiologic changes, as I described, that occur in pregnancy can definitely alter the metabolism of those medications. So certainly we could do it. I think the issue really is going to go down to funding and who is going to support getting those studies done. And going to Susie's point of real world data, so some of those studies are done, right? So sometimes we have patients who come in with epilepsy and they are on an anti-epileptic and they are still seizing. And so we get drug levels on them, right? So could we pull that information and be able to then give us some information in pregnancy for them? And is there a way to get everyone's electronic medical record labs pulled out to be able to get that information and get the clinical data to match it? I mean, is it possible? It theoretically could be possible, but it will be a big undertaking.
I would just, add to that. I think one of the things that when we think about sort of the real world data piece, so the electronic medical records is super important. I think we all think it's super easy, right, in theory to do, but once you get into the details of interoperability and even, you know, mom's records don't match baby's records. So how do you follow up on that? So that's a real challenge. I think the other challenge when we think about sort of the registries is that they all kind of work a little bit differently. And so some of the challenge for, if you're a provider or a patient and you see that there's a registry, which it'll say on the label, which is great, right, that it exists. But if you go try to find it, you may not have access to that data. It may be, I think an example is several years ago, we were looking up something as an example and it said, oh, we have a report on this. Please call us to let us know if you would like the information. That's not terribly helpful when you're trying to decide sort of a course of treatment. But I think there are things that I know FDA has done some work on the pregnancy exposure registry side of things that I think will be really helpful. And so thinking about those types of opportunities, but the data piece and who owns it and how people can access it is also a challenge as well.
Christina, you just beat Maggie out there. So Maggie, you're next.
Sorry, Maggie. I'm sure you'll say something brilliant as soon as I'm done. So, and to get to that question about PK and then also to Katie's comment about registries, I just think there's smarter ways we can deal with the data that we're already generating. So for example, in our clinical trial programs, and someone mentioned it, and I don't remember who it was and in which session so far today, that while there may be exclusions for pregnant women in trials, we do rarely see patients become pregnant in our trials. And we're missing opportunities to collect information. The patients already consented for participation in the trial. They wanted to be in the trial. They wanted to contribute their information to science to help either benefit their situation or someone who was following beyond. I don't think we're taking proper advantage of understanding our mechanisms to know when it could be appropriate that that patient could continue in the trial and we could generate some simple PK data that could inform on some basic information about appropriateness of dose in different trimesters of pregnancy. So I think that's one low-lying fruit solution that we just need to be smarter and really pay attention to the science we already have to inform on some of these decisions. And then the second thing about registries, and it won't be a surprise because I've said this publicly in a lot of different forums, when we're interacting as sponsors with the agency and the agency's interacting with us, we are appropriately, because this is the way we're required to do so, having a conversation about one product at one time. But we're in a situation here with pregnancy where it's not appropriate to have a drug-specific registry that provides limited use at the end of the day. So I think that there needs to be broader collaboration when we talk about public-private partnerships to really establish and entrench within the system pregnancy-specific registries for patients who, when a product is approved and it's not contraindicated for use in pregnancy, that those patients will automatically be enrolled in a registry where we will collect information to help better inform on the appropriateness of dose across the different phases of pregnancy and the safety information. So I think there's just different ways we need to think about the way we're already seeing patients and already have opportunities to capture information that would provide robust sets of information to better inform about our products.
Thanks, Christina. Maggie, you were next. And then I'm going to ask one last question to everybody, and I'd like everybody to weigh in. I know we're running out of time a little bit.
Yeah, two quick things, other than plus-oneing what everybody has just said. So what's really important when we're getting real-world data is prospective and having the denominator. Other just sort of case studies can actually, are they sort of useful? Well, sometimes kind of, but often they are the worst thing with research with pregnant women, because some bad adverse events have a high baseline in pregnancy. 3% of pregnancies end with fetal abnormality, just as a baseline. Early loss, high baseline, right? So if you just say, oh my gosh, this woman was on a drug and then looks, this terrible thing happened, and do case reports in that sense, it actually is deeply problematic. So we need representative, high numbers, but we need prospective with denominator. The second thing is, I don't want to end on a bad note, but I do want to just a little caution on electronic medical records. We would need to remember that the broader social context and political context with pregnant women get surveilled and jailed, especially women of color. When we find out that something happened to their baby, there's an enormous tendency, if there's any medical evidence that she took a drug or something, pregnant women have gone to jail for this kind of stuff. So we need to have, as much as we want to leverage the promise of electronic medical records, which I think we should, better have the feminists work out the structure. That's what I'm going to say.
And I want to apologize up front for all of the excellent questions in the Q&A box, and hopefully what we can do is transfer some of them over to the further discussion tomorrow. But I want to end on this final question for everyone to weigh in on, and that's what strategies could be used to advance the culture of including pregnant people in research? Okay, someone has to. All right, Christina.
Sorry, I feel like I've spoken too much, but I think it's quite straightforward, and I mentioned this in my prepared comment, that we have far too often just had a standard exclusion in a protocol. And I think we are beyond the time where sponsors, where IRBs by regulatory agencies need to be asking what is the basis for that exclusion and what is the science to inform that exclusion. It's such a simple fix. Every protocol we write is reviewed by it has to go through a clinical trial application with every country where our trials are going. It's a simple way for us to change this culture of inclusion.
I want to just echo that. Absolutely. I think people need to be held accountable. And right now you're not. It's not at all. If you exclude them, that's the norm. And we need to move beyond that. And we need to be accountable to why they are excluded, why pregnant and lactating women are excluded.
And I would just add that I think that it's almost novel at this point, but should have been, for centuries, that pregnant women can make their own decisions. And so autonomy and the ability to participate if you would like to, rather than being excluded at the get-go, I think that's going to be really important as we look to clinical trials in the future.
Anyone else? Yes, sorry, Christine.
Hi,
Sorry.
Susie.
Yeah.
I just want to give credit to Cynthia. The one thing Cynthia said that I just love is you tell women that's probably safer for them to be in the trial than not, where they're just an N equal 1. If they're just getting a COVID vaccine, that's a case report, right? And we don't know what to do with those data. So the whole point is really reframing the message that's been in place since thalidomide. I mean, we are still living in the shadows of thalidomide. We haven't moved from there. That's why we're having this workshop. We need to move away from that. So it is pointing out the danger of not being in a trial and the advantages of being in a trial. I mean, there is all sorts of unknown in a clinical trial. I mean, to every subject who gets signed up, right? And if the pregnant person can make that informed consent, and if we think the available data is such that it is reasonable to enroll pregnant people, then that's the scientific basis. We are currently operating out of a base of fear. That's why pregnant women or pregnant people are just automatically excluded. Why is that? Because we're fearful. And that's not how we do science in 2021. So I think capturing some of the ideas that Cynthia brought up, because you're in the throes of it, would be a great starting point. And this has to be done certainly at the community level, at the provider level. I will point out everything pregnancy related is controversial. Everything. Even when it is the most straightforward thing, it is controversial. So I can appreciate this, this enthusiasm, this need, based on need really, to enroll clinical people in trials when it's necessary. That messaging, I'm sure could be counter with differing opinions.
So, Christine, you're going to have the last word on the panel. I'm sorry, because I could listen to this panel talk for the next 2 hours. It's been just such a joy to moderate this session, and great to see all of you, and thank you for all of your input. I'm going to do a little bit of a wrap up, and then we're going to let everybody go. First, many thanks to all of our presenters, panelists, and other experts around the virtual room for helping us to host such an engaging discussion throughout the day today. I think we managed to cover a lot of ground. We look forward to making good use of all of the feedback that we gathered today. And in terms of just a brief summary of where we were today, Kavitha Vashist introduced us to all of the activities in the Office of Women's Health at the FDA and highlighted the need for the discussion today. And I think it's just been an incredibly robust discussion. In the first session, we discussed the need for participation of pregnant people in clinical trials and efforts in the area of FDA guidance, the PregLac task force recommendations, and the implementation plan for the PregLac, as well as the clinical needs to optimally care for pregnant people, including including information to support safety, efficacy, and dosing. And then I just wanted to highlight the first session by the statement that pregnant people deserve evidence for prevention and treatment of their illness equal to others, which I think is just really important. In the second session today, the speakers presented information on the types of non-clinical studies used to support clinical investigations in pregnancy, and that information was summarized. There was information on the interpretation of the data and how it is used to characterize risk and ultimately translate into labeling statements. Non-clinical reproductive toxicity studies can provide information to help identify and characterize the potential risk to pregnant individuals and their fetuses, and allow investigations in those pregnant individuals. And then a summary of all the available non-clinical data are considered when characterizing the risk. And then just to reinforce that sponsors are encouraged to talk to the FDA about the non-clinical program needs to support enrollment of pregnant individuals in clinical trials. And then in the final session, we really started to get into some of the specifics of designing clinical trials that enroll pregnant people. The focus of the session was, and I love it, my dog is singing to me behind me. I hope you can't hear him. He thinks he never gets fed. He's already had 2 meals today. Sorry. So we started to get into the specifics of designing clinical trials that enroll pregnant people. The focus of this session was to discuss the ethical and scientific challenges when considering the use of investigational drugs, factors that impact decisions to evaluate specific therapies in pregnancy, including ethics, unmet needs, the benefit risk of using a drug and the risk of having an untreated condition. And the panel really discussed the potential designs for trials in pregnant people, including some of the dose finding, talking about some of the pragmatic trials, the opportunistic PK studies. We didn't really get into some of the natural history studies, but I think talking about real world data gets at some of that. And then really focusing on efficacy and safety studies. We did touch on the use of real world data and some of the challenges associated with that. You also discussed the timing of enrollment of pregnant people to address some of the physiologic changes. I'm hoping we'll get to a little bit more of that tomorrow, but specifically talking about the potential need for different dosing strategies throughout the course of pregnancy. It was discussed in terms of the need for endpoints for both the pregnant person as well as the fetus, a discussion of infrastructure needs such as what was presented for the MFMU network, and then a final discussion about respecting the autonomy of the pregnant person and really working with them to be able to provide them with what they need. So I'm going to stop there. I'm going to close the meeting for today. I want to thank everyone who joined today. I want to really encourage you to come back tomorrow because we're going to talk about case studies and a discussion of the challenges and next steps and how we implement what we talked about today. And then just as a reminder to all the speakers and attendees that you should use the same link that you used today for tomorrow's meeting, and that we will see you tomorrow at noon for the next phase of the workshop. And I want to thank you, and I look forward to seeing you all at noon tomorrow. Thank you.